Good afternoon, everyone. Thank you for joining us today on day one of our two-day conference. Please let your colleagues know that we have, they could register to get in today or next Thursday. We'll be here again in the afternoon. Some housekeeping. We have a couple of sessions today and then next week, and each one will have a keynote speaker or a group of panelists. If you would like to submit a question, please use the Q&A feature on the bottom of the screen. If you want to be anonymous, just choose that option. And if you have technical issues, use chat and we will get you through them. We've had a few technical issues, as we always do, but we got through them. So I am Sheila Garrity, and I am very pleased to introduce my boss, Pam Norris. Dr. Pamela Norris is the Vice Provost for Research at George Washington University, where she reports to the Provost and Executive Vice President, serving on his leadership team. The Office of the Vice Provost for Research at GW works to strengthen existing initiatives and expand into new multidisciplinary fields. The office works in tandem with faculty to facilitate all phases of the external and internal funding processes and support faculty and students' scholarly initiatives, providing leadership around new ideas, insights to key leaders, input and feedback on initiatives, and championing programs that promote research and scholarship across the university. Dr. Norris joined us in November. Previously, she served in roles as the Executive Dean, the Executive Associate Dean of Research, and the Associate Dean of Research and Graduate Studies at the University of Virginia School of Engineering and Applied Science, and is the Frederick Tracy Morse Professor Emerita of Mechanical and Aerospace Engineering. She's a native of Portsmouth, Virginia. She joined the faculty at UVA in 1994 after receiving her undergraduate degree from Old Dominion, her MS and PhD degrees from Georgia Tech, and completing postdoc studies at UC Berkeley. She's recognized globally as a leading expert in nanoscale heat transfer, especially interfacial thermal transport, with a focus on thermal management across a range of length scales. She holds patents for innovative thermal management techniques for jet blast deflectors, as well as for applications of aerogels in areas ranging from biological warfare detection to lab on a chip to thermal insulation. She has served as the PI or co-PI on over 45 sponsored research projects, representing well over 25 million from DoD, NSF, industry, and foundations. Dr. Norris is well known for her mentoring skills and for her dedication to increasing diversity in the STEM disciplines. In 2016, she was honored with the Society of Women Engineers Distinguished Engineering Educator Award for enduring positive influence on students' lives as a gifted teacher, mentor, and role model, and for promoting greater diversity in STEM higher education. She is also well known for leadership in the field of nanotechnology education, chairing the American Society of Mechanical Engineers National Nanotechnology Institute's Committee on Nanotechnology Education from 2003 to 2010, and organizing the first national nano training boot camps at the leading edge of the field. In 2021, she was elected an honorary member of ASME for international leadership in nano, micro, and macro scale thermoscience and engineering research, for tireless efforts to advance diversity in STEM fields, and for demonstrating engineering excellence as an outstanding mentor for students and faculty. She currently serves as the editor-in-chief of Nanoscale and Microscale Thermophysical Engineering, and recently served as the Vice President of Institutional Councils for the American Society for Engineering Education and as chair of the Engineering Research Council. And I got to tell you that we put Pam through her paces. Her very first day at GW, we had a bunch of snafus getting the computer up and running, and here we were today trying to get her on screen. But she takes it all with a smile, and I feel smarter just reading her bio to you. So I am going to mute myself, Pam, and turn it over to you.
So Sheila, I was just going to say, I'm not sure which was more painful, trying to get into Zoom this morning or listening to that extended bio. It's always so difficult. But welcome, everyone. It's so nice to have you here at the 2022 DC CTSA Spring Reading. I want to thank our event partners at the Georgetown-Howard University Center for Clinical and Translational Science and the Clinical and Translational Science Institute at Children's National. I also really want to thank our event organizers at GW. We got Sheila Garrity, Hiromi Saunders, Mariel Heiss, and Rebecca Eberly. At Georgetown, we have Mary Shindell and Sarah Vatone. At Howard University, Jane Ottawato, and at the DC, Virginia Medical Center, Priscilla Adler and Claudia Ganawan. And I apologize if I mispronounced any of those names. That was an interesting array there for me. This marks the third time the spring meeting has been held virtually, I'm sorry to say. But remarkably, in 2020, this event shifted to online with only a week's notice. Amazing. It's really sad, though, that for 3 years we've had to do it this way. And I know we all look forward to gathering in person. But although we can't be together in person, I want to commend our organizers for their commitment to this meeting. The watchword for this particular event and the clinical research enterprise overall, I would say, is collaboration. Translating discoveries from the lab to the clinic requires the expertise and efforts of countless individuals. Whether you're working at the bench or you're writing and reviewing protocols, your contributions are critical to the innovations that will advance our understanding and treatment of diseases. So today's theme is the lasting impacts of COVID. We've all personally been touched by the toll of this disease in one way or the other, and will continue to be impacted by it for the foreseeable future. Shortly, you'll hear from Tim McCaffrey, a professor in GW School of Medicine and Health Sciences. Dr. McCaffrey will talk about his work to enable faster and more accurate testing for COVID. He'll discuss a technique for identifying biomarkers of the infection in the bloodstream. This novel approach could produce new ways to test for numerous diseases and disorders, a wonderful side effect of this pandemic. His work really is just one example of how the pandemic has required many of us to rapidly pivot in a different direction, to learn everything we can about this virus and to work to prevent the spread of coronaviruses. And the lessons we learned will undoubtedly inform other areas of research. We've innovated on the fly. Funding has been boosted for COVID-related research projects. Our institutions have prioritized protocols that have anything to do with the disease. We've tested and launched new models of PPE, of vaccines, and of clinical treatments. And of course, all of this was possible thanks to the dedication and hard work of the people in this Zoom room and your colleagues across the country and the globe. Our theme on May 5th will be artificial intelligence in research in healthcare. On that day, you'll hear from Dr. James Garradano. I think I got that right. A professor at Georgetown. He'll be talking about emerging trends in AI, specifically the ethical and legal challenges that AI poses. AI is being developed at a very rapid clip, and if we're not careful, it stands to recreate, or perhaps even worse, accelerate the inequities and biases that already currently exist. So preventing those consequences is near and dear to us at GW. A team of researchers, in fact, in our School of Engineering and Applied Science recently received a $3 million grant from the National Science Foundation to create and launch a training program for future designers of trustworthy AI. These graduate students will be taught to design AI that takes full advantage of the capabilities for social good while mitigating the downsides. And GW has also joined Howard and Georgetown in the Public Interest Technology University Network. The mission of that network is to create a new field of public interest technology. one that really centers on people and the development of technology and related policy making, a field that critically assesses the ethical, political, and social implications of these advances. When this conference started, it was a regional conference, I understand, but joining us today, because we're hosting this online, there are more researchers and research administrators that are joining us from outside of the DC Capital Region. We're thrilled to have you with us and to join this conversation. Between our speakers and all of your expertise, we're looking forward to very lively discussions. I thank you all for attending and for participating. And with that, I'll turn it back over to Sheila Garrity to introduce our first speaker.
Thank you, Pam. And I have to tell you, as many of you in the audience who have participated in conference planning, when the day comes, it's kind of like when you're planning a party at home and are all the details ready, and here we are. We're so excited to have you here. We worried that we couldn't pull it off, but we know it's going to be a great event. Every day after the conference, today and next week, you'll receive an evaluation form, and I would please ask you to honestly complete that so that we can use the feedback for next year. Our colleagues in the district at Howard and Georgetown and DC VA, we're a model of collaboration. We've broken down walls, and as Pam likes to remind us, we all have a part to play in this research enterprise. So thank you for being here. And Hiromi Sanders, I'm going to turn it over to you to introduce our first speaker.
Thank you, Sheila, and thank you, Pam, for that welcome. I have the honor of moderating today's keynote address by Dr. Timothy McCaffrey. Let me read a little bit about Dr. McCaffrey. His scientific interests involve investigating the molecular mechanisms of cardiovascular disease. His lab employs genomic methods to identify the causal connection between immunity, vascular inflammation, and the atherothrombotic process. His lab's highest priority is moving forward with the use of next-generation RNA sequencing technology to provide a high-definition view of the transcript differences in blood RNA associated with the presence of coronary artery disease. Further, it will provide interesting new leads about the etiology of the disease and possibly new therapeutic targets. If successful, digital RNA quantification methods opens up an exciting platform for diagnosing other devastating cardiovascular events, such as heart failure, stroke, and aneurysm before they occur. Dr. McCaffrey received his BA from St. Mary's University and his master's and doctorate from Purdue. He received postdoctoral training and later founded the Genomics Core facility at Weill Cornell Medical School and served as Director of Research in the Division of Hematology Oncology. In 2001, he relocated to the GW Medical Center, where he co-founded the McCormick Genomics Center. He was awarded a prestigious Merit Award from the NIH for his work on the molecular genetics of vascular aging. He serves as an associate editor for the journal Gene and is a director of the Division of Genomic Medicine at GW. He serves on the board of trustees of the Heart and Vascular Institute at GW and is a member of the Science Advisory Board for the Katzen Cancer Center and is a member of the Expert Advisory Panel of the Rare Genomics Institute. The title of his keynote address is Transcriptome Profiling of Human Blood as a Platform for Diagnostic Biomarker Development. Measure broadly, listen carefully. And with that, Dr. McCaffrey, thank you for being our keynote speaker. The floor is yours.
Thank you very much, Hiromi and Pam, for the introduction. I guess I'm gonna have to try and do a good job now. That's let me bring up, with any luck, let me bring up the says I'm screen sharing. Do you have my slides in front of you?
We do, but they're in presenter mode, Tim.
Gotcha. Okay. There we go. That should be full screen.
Yep.
Great. Thank you very much. Just note at the bottom, there is a disclosure there as a function of what we do. We've more or less had to start a spin-out company, True Bearing Diagnostics, which is commercializing these RNA-based diagnostics. I'm going to invoke to put a frame around this, I'm going to invoke Niels Bohr, who defined an expert as somebody that has made all possible mistakes within a narrowly defined space. So buckle up, because I'm going to tell you about kind of all the mistakes that we've made over the last 2 or 3 decades. And we made those mistakes so that you don't have to. This is what we teach. This is medical genetics 101. This is what we teach to first-year medical students. It applies to almost all diseases. We think it's kind of based on the Vogelstein model, a 2-hit model of a gene by environment interaction, and that ultimately predicts your risk of disease. As you know, we've known about heritability through DNA since kind of the 50s, Watson and Crick, but we haven't really had the ability to measure DNA very well until the Human Genome Project. The effect of that is that we have conflated heritability of a trait, let's say a disease trait, we've conflated that with DNA because we haven't really been able to measure the DNA very well. Well, now we can, and I'll show you what those methods are. And then the kind of interesting part of this, the more and more we measure variations in DNA, and the more that we now full-on sequence people, we realize that the DNA variations do not account for as much disease risk as we thought. In this conventional model, you'd have variations. You'd have 2 alleles, a maternal and a paternal allele that here we're calling G and A. Those lead to gene expression in genes that are coded, that have exons and code a protein, and that then changes the function of the protein. And then presumably in our hypothetical model, that explains why somebody that could have bad genes like Jim Fixx, that was a marathon runner but died of a heart attack at 52, versus somebody that's got great genes, Winston Churchill, arguably the most stressful job in the world, clearly did everything wrong, smoked, drank, stayed up late, the whole 9 yards, and yet lived to be 90. Those good genes then interact with our environment, our diet, exercise, stress, etc., and that gives us our actual complete sort of risk of disease in this model. Problem is, I'm going to show you very, very briefly in the next few slides that it's unfortunately kind of wrong. Where I've been focusing for the last 2 decades, and I'm going to continue to focus, is on gene expression. And it's a lovely entry point because it clearly results from heritable DNA changes to some degree. But gene expression embeds something very, very different, very, very important, which is epigenetics, both at the methylcytosine level, at modification of the DNA itself, and then modifications of the chromatin, of histones and things that modify the accessibility of those genes, ultimately determining which genes we use in a particular tissue or disease. Well, gene expression also embeds and reflects our environment, and it's directly modified by the proteins that we make, transcription factors, RNA binding proteins that we'll be talking about. And we've done a lot of trials. We've done a lot of clinical trials. This is actually just a short list of trials we've done. With our cardiologists, we've looked at Herceptin, adriamycin cardiotoxicity, heart failure, and the way that that is reversed by left ventricular assist devices, obesity, bariatric surgery. We've used DNA sequencing to identify pathogens in ICU patients by using 16S sequencing. Aspirin resistance in high-risk coronary patients. We just finished a really interesting study, I'll tell you about some other time, on identical twins that are discordant for ADHD, and did RNA sequencing of their blood to identify potential biomarkers for it. So today, believe it or not, we are going to talk about COVID, but I'm going to explain a little bit of a roundabout way that we got to COVID, because I think it's fair to say nobody exactly saw that coming. We were working on other infectious diseases. We're working on the diagnosis of abdominal pain, particularly appendicitis, pneumonia, and biofilm infections. And actually, my core work is on the diagnosis of coronary artery disease. So that's where we're going to start, just very briefly. As I alluded to, the ability to measure DNA has been expanding vastly faster than Moore's law on computers, to the point where the first genome, which is in effect Greg Netter's genome that was sequenced, probably cost $3 billion and took 10 years. We can sequence a person overnight for about $300 now. The big jump in this came in about the year 2000, when it was figured out that you could print arrays that had small features on them, and each feature, as you zoom in on them, each feature was about 11 microns in size, which is only slightly larger than a red blood cell. And each of those positions on the array could have a particular DNA sequence on it. That DNA sequence then could capture labeled DNA from a person or labeled RNA from a person. It really didn't care. And you could use that to interrogate, to guess, if you will, what the person was, what their DNA sequence was, or how much RNA they had. And then each of those, you could read each of those pixels, and you knew what was in the pixel by virtue of its XY coordinates in the array, hence the term microarray. These were incredibly powerful systems. So we hammered these and applied them to any disease that got in our way from about the year 2000 through 2010. They were used to interrogate single nucleotide polymorphisms, because you could put, at the time that this slide, 2007, that it was made, there were 500,000 variations in DNA that were known. And you could interrogate those variations in DNA across the entire genome. And these are very large studies. So this is 14,000 patients that are then split into various groups. And on the panel here, you see that the chromosomes are just numbered from largest to smallest. So chromosome 1 is your largest chromosome, over to 22, and then the X chromosome. And each position here is vastly, vastly crammed down. But those 500,000 positions across the genome are then examined for a C or a T, whatever the variation is at each of the polymorphisms. And then when you divide it across the group, you say, well, is that C allele, is that C variation disproportionately represented in people that have bipolar disease, or in the second panel, coronary artery disease, Crohn's, hypertension, et cetera? You get the idea. Well, the point of the story is that as you get up to about 3 on this log scale, you're actually just talking about random noise in the genome. And so we know that bipolar disease, for instance, is a highly heritable disorder, and yet I kind of hate to break it to you, there's no good gene for it. There's no variation in DNA that particularly accounts for your risk of getting bipolar disorder. Now, in the second panel, you see there is one transcript shown right here, and it's on chromosome 9 at a position called 9p21. And to this day, that's what the gene is called. And it turns out it's a non-coding gene. It turns out it does not code a protein at all. It encodes an RNA that is circular and is regulatory to other RNAs. But it already starts to violate that model that I just got finished showing you, whereby a DNA variation is supposed to change a protein, and that causes disease. This particular locus for coronary artery disease at 9p21 yields, as it shows on the left over here, about a 1.6-fold risk of a heart attack. So that makes it equivalent to, say, having high cholesterol. You can have a bunch of these. You can have, we don't even test, honestly, for 9p21 in our coronary patients. We do, of course, test for cholesterol. You test for hypertension and smoking, and you know the risks. But the important detail is that when you add up all of those known risks for coronary artery disease, you only explain 40% of the cases of heart disease. The effect of that is the vast majority of people who have heart attacks are taken by surprise. They are as surprised as anybody that they had a heart attack. They don't drink, smoke, they exercise, etc. The study that I'm going to show you today is people that don't have an acute myocardial infarction. We base this study on people, it's a little bit dramatic here on the left panel. We're basing it on people that have what are called stable chest pain. So they go out and exercise and their chest hurts. They have the good sense to go to their cardiologist and get a workup. But even if they present at the emergency room, only 11% of people with chest pain end up being diagnosed with coronary artery disease. The vast majority of people with chest pain have acid reflux. You can have a pulmonary problem, you can have a pinched nerve, you can have stress in the intercostal muscles. But you do need to check for it and you do need to rule it out. The method of ruling in or ruling out chest pain as coronary artery disease is done by a stress test. What our goal here is, our high hope is that we can make a blood test, and I think we're very, very close to it. If you fail your stress test, you get EKG changes, and first of all, don't be alarmed. Practically everybody fails the stress test. You're likely now to end up with a CT angiogram or a calcium score. But at the time we started these studies in about 2015, you were very likely going to get an invasive coronary angiogram in which a dye would be flowed through a catheter into your coronary arteries. And in the lower right here, I'm showing you normal coronary arteries, a nice perfusion of the heart. And then conversely, you see that when blockage occurs, you get a really nice imaging of where that stenosis occurs. The beauty of this is you're already on the table. They can thread a stent in there, open that up, and the chest pain will alleviate on the spot. So we know that this stenosis of these arteries causes chest pains. But the kind of lesser known fact is that it actually has almost nothing to do with the disease, and it doesn't change your risk of dying of a coronary event. So it alleviates the symptoms, but it doesn't really change the disease or the course of the disease at all. Now, another thing that I need to stress at this point is that even though you may image somebody and they have what appear to be patent open coronary arteries, you actually do not know that they have disease that is not occluding the artery. And by doing CT angiograms now, we know that that reflects about 10 or 20% of cases. So kind of put a pin on that and we'll come back to why that's important. Our strategy that I'll throw open can be applied to almost any sort of diagnostic procedure you want to make, is that we pull blood from these people at the time that they go in for the angiography, and then we get a result from whether they have blockage or not. We get a result within a matter of hours. So we can preserve this blood, we can file it away, and then we can subject the blood to the microarrays as the way we started, and I'm going to show you RNA sequencing. And then we simply divide our patients into people who have coronary disease and those who don't have coronary disease. And we look for transcripts that are informative or diagnostic of that difference. So I showed you the microarrays before, and in those microarrays, each of the features or pixels that I showed you was about 1 million copies of a particular DNA sequencing. Well, over the subsequent 10 years, friends of mine at Sequel and other companies also figured out how to image individual strands of DNA. And so now the pixels that I'm showing you here are individual strands of DNA that are attached to a glass plate. And if we zoom in on just one region of that and focus on 3 of the spots, I'll show you how we sequence each of those strands. What you do is you flow in just one nucleotide. So you flow in a G, and the G is fluorescently labeled, and so it incorporates at position 3 here, but you see position 1 and 2 are not incorporated, so they're not scored. You erase that fluorescence, and now you flow in a C, and it incorporates at positions 1 and 2, and you erase it, and you flow in an A, and you flow in a G, and so you flow in all 4 nucleotides, and you keep doing this, and then at each of these positions, you're building up DNA sequence for each of those strands. Now, what's impressive about this technique is I'm showing you 3 positions here, but the technology routinely tracks 50 million positions on 50 separate channels. So we can get 50 million reads from 50 patients in about a 4-day run. That gives us about a gigabyte of sequence from each of those patients. I'm going to leave out the magic, the voodoo computational things that intervene, but we take those sequences, we align them to the human genome, and then we count them into whatever transcript is made from that part of the DNA. So we basically count them in to the individual transcripts. In this particular case that I'm showing you here, we're counting them into 157,000 transcripts, which is an incredibly precise way to do it. We only have about 28,000 genes, but each of them has different splice variants. And so that's where you get 150,000 transcripts, roughly. What is important about this is if you just look at it at a meta level, we're looking at each of the spots here, each of the points. Let's take the topmost point in the upper right. We're looking at its expression level in people who do not have coronary disease versus people who do have coronary disease, where more than 20% of that artery is occluded. In those cases, I can tell you what this transcript is. This transcript is globin, which is, of course, in the red blood cells. And fortunately, that transcript does not change at all. It's expressed at a very, very high level in blood, but it's expressed at exactly the same level in both conditions. So it's unchanged. Simply put, all of those things along that line are uninteresting to us because they don't change between the 2 disease conditions. Where you start to get interested is when you get down into transcripts that do differ statistically between the 2 groups, and those are shown in black down here. The important feature of this is that all of these transcripts now that we're identifying by RNA sequencing would not have been detected by microarrays. They simply aren't sensitive enough. The elaborate proof for that argument is that there's actually a company called CardioDx that identified markers by microarrays and even took it up to the FDA and was selling the test, and it proved not to be useful. So we know that the microarrays did not give us the correct markers. We feel pretty confident that these do. Now, I'm not going to go into it, but the real power of this technology is not even shown here. Its quantitative ability is stunning. These are log 2 orders of magnitude. So we're quantifying from less than 1 transcript per cell up to about 65,000 transcripts per cell. But you can drill down even further, and you can take any one of those transcripts, and as I alluded to, you can look at the way the reads that you get tell you about the splicing of that transcript in relationship to disease. You can get the single nucleotide polymorphisms, so you can get the genotype of the patient as well. And then if they differ, if for instance they have a CT variation at any given position, you can determine which allele that they're using. Now, let me just pause for a second and tell you something kind of useful, which is that when I showed you those genome-wide association studies, kind of often fail to find an informative gene. The problem with them is that they assume that you're using each of your alleles, your maternal allele and your paternal allele, equally. That's a core assumption because they can't tell which one that you're using in your brain or your heart or your blood. Well, we can. And what we can tell in study after study is that, yes, you got a C allele from your dad and a T from your mom, but in your T lymphocytes, you might only be using the maternal allele. And so that would then throw that whole genome-wide association assumptions into complete disarray, no pun intended. So now I'm just going to show you that this particular pattern that we got was, it was downright upsetting, I'll be honest, for one particular reason. We've done 50 studies, and in every study, when you tend to get some transcripts that go up in the disease condition and some transcripts that go down in the disease condition. Some of that is noise, but also some of that is real. When we found transcripts that changed in coronary disease, they all went down. And they all went down by almost exactly the same magnitude, which is about 1.6, 1.7 fold, regardless of their absolute level of expression. And that befuddled us. And probably did a month or two worth of head scratching before we figured out what that pattern meant. And I'll tell you in just a minute. The pattern was reproducible. So on the left is the study that I just showed you, what we call the TRACE-IT-1 study, that was patients from GW. And there were 48 patients with and 48 patients without coronary disease, a total of 96. We did simultaneously a study in Argentina for a related reason. We call it TRACE-IT-2. The pattern was the same. All of the transcripts in people with coronary disease at about a 9-to-1 ratio were in the downward direction. Again, very puzzling. And now a third study that we just finished about 2 years ago with our colleagues over at Innova shows, again, very much the same pattern. When you look just at the genes that are changing, they are vastly in the downward direction. Well, before I tell you what they are, let me just tell you that they're useful. So for those of you that are into clinical diagnostics, you know, and I'm sure whatever your specialty is, that you have clinical diagnostic models that use things like age, gender, now it's sex, symptoms, diabetes, whatever the known risk factors for the disease are. There's typically some weighting scheme. We're using one here called a diamond scheme. And when you accumulate those risks between the two groups, without CAD and with CAD, there is statistical significance, barely, between the two groups. It is slightly predictive. But by comparison, if I take just 7 transcripts from the roughly 200 transcripts that I showed you, and do the same thing. And I told you that the transcript is always in the downward direction in people who have coronary disease. And if I just do a simple additive model of those 7 transcripts, I get obviously a larger difference, like I said, about 1.7-fold difference, but I get a p-value of e-12. It is wildly stronger as a predictor than the known clinical risks that we have. Let me pause for just a second and tell you what not to do here, which is it is very attractive and is built into all the analytical software, and it is now built into almost all the publications that you're going to see, which is to do something like take those 200 transcripts that differ and use a machine learning model, use a neural net, use a partial least squares prediction model that takes those 200 transcripts and groups them and weights them and squares them and comes up with a prediction model. I guarantee you, you can predict 100% any disease from any group of variables that you get. The problem is, as soon as you go to a different group of people, it completely falls apart. And the term is overfitting. You can take a bunch of variables and you can fit anything that you want. If you want to listen to my experience on this, don't do that. Investigate, track, listen to the particular transcripts, find out what they're doing, how they're related to the disease process, which I'm going to show you here, and then pick ones that are connected mechanistically to the process that you're trying to diagnose. So for those of you that are into clinical diagnostics and into receiver operating characteristics, the clinical model, and this is well known, has an accuracy of only about 54%. So in other words, you're roughly better than flipping a coin on an ROC curve, that's this red line here of 0.5, there is some information embedded in that, in your trade-off between your true positives and false positives. But if you look at our RNA-based model, our ROC is about 0.873. We can get up into 80, 85% sort of precision. Now, and this is where I need to remind you that I don't think we're ever going to get above 90% accuracy, because remember, I told you that people that have apparently clean arteries by angiography might actually have coronary artery disease. And the transcripts that we have are sensitive to the process of atherosclerosis. They are not sensitive to the stenosis. So until we go to something like CT angiography, I don't think we're going to get much more precise than about 85, 90% max. Well, another thing that you got to think through is that you may do all of this work and then come out having just diagnosed something that's true but uninteresting. What if, after going through all of this, our markers were simply markers of cholesterol? So you got to go through all your known covariates and figure out whether your transcripts are sensitive to that. Just going to give you the short story here using two different methods, one a statistical method, and then one to go and find the transcripts associated with smoking or high cholesterol or aspirin use. Our transcripts are not sensitive to any of those known covariates. They appear to be predicting something completely different about the coronary artery disease process. And now I'll tell you what it is. So we were perplexed by how those transcripts always went down, and they always went down by the same amount. There was a kind of a spooky similarity in the magnitude of their change. And it dawned on us that what was happening is this was probably reflecting a particular cell type that was at a lower level in people with coronary disease than in people who didn't. So we went to the literature, and you go to the literature, and the first thing that you find is about 100 or 200 papers suggesting that people that have coronary disease have lower levels of different types of circulating progenitor cells. And we can go and find those transcripts in our data, and it's just, we can't defend that position. We can basically falsify that it's progenitor cells that are differing. But then the next largest literature that you'll get is on the role of T regulatory cells, particularly in the T cell family, that are again known to be at lower levels in people that have coronary artery disease. So we could take transcripts that we know are representative of T cells, granulocytes, lymphocytes, etc., B cells, and then we can compute a composite level for those transcripts across our groups of different coronary artery levels or different CAD levels. And what we saw started now to make sense. The people that had mid to high levels of coronary artery disease had lower levels of transcripts for T cells, not for granulocytes, neutrophils, somewhat for lymphocytes, particularly CD8 T cells, but again, B cell transcripts completely unchanged. So we couldn't falsify the theory. This is just showing you that it is an incredibly reproducible phenomenon. Every major cath lab in the world that's looked at it finds a Th17, a Treg imbalance in people with acute coronary syndrome, with various types of coronary atherosclerosis. And I'll take just one example here to make an important point. The other question is, well, okay, maybe it's true, but if it's true, would it explain the magnitude of changes that we get? This is just one of the publications where we could parse out what the reduction in T regulatory cells was. And assuming we don't have coronary disease, you and I have about 5.3% of our lymphocytes as T regulatory cells, whereas in people with coronary disease, that drops to about 3.4%. When you compute that out, that would predict that there'd be about a 1.6-fold change reduction in Treg related transcripts. And as I showed you before, that's almost exactly what we get. So it is at least consistent with the idea that changes in T regulatory cell numbers could give us the transcript profile that we see by RNA sequencing. Importantly, it is not a coincidental relationship. We won't be able to go into all of this, but this is lovely work from Renee Vermani out at Uniform Services, showing that the T cells are actually intrinsically related to the stage of atherosclerotic progression. And overall now, when you look at the relationship, the known relationship between Tregs and atherosclerosis, it's a very compelling argument. We know that Tregs go down as a natural function of aging. There's probably just some degree of senescence, fatigue. Some people use the word exhaustion. Tregs are reduced by hypercholesterolemia. So again, that's a fit. Really interesting is that statins used to treat hypercholesterolemia increase Tregs in patients, and it's possibly part of the bonus effect of statins that you get an increased benefit disproportionate to how much you change cholesterol. Well, again, in terms of causation, in animal models, if you force Treg deletion in monoclonal, in transgenic mice, you favor atherosclerosis in those mouse models. And then far and away, the most compelling argument for a relationship between immunity and autoimmunity and coronary disease is the well-known relationship between disorders like lupus, psoriasis, rheumatoid arthritis, and their known association with coronary artery disease. I was kind of unaware of this, but the reality is that people with lupus, the major cause of death is actually coronary artery disease. So there's good sort of face validity. There's a good fit between the immune system and the formation of coronary disease. The particular markers that we use, and this is my argument about profiling broadly, but listening carefully and trying to figure out what the transcripts are doing. I spent easily a year figuring out by various techniques what these transcripts were doing. And they're shown, the 7 that we think are the best, SMID3, TCF3, IKZF4, which is called EOS, are involved actually in the transcription of FOXP3. Well, FOXP3 is the absolute consensus marker and is mechanistically involved in the differentiation of T regulatory cells. FOXP3 is itself a transcription factor that binds to 2 other factors that we're using, transcripts that we're using, TRIM28 and IRF4, and that helps to regulate the transcription of these FOXP3 target genes. Those target genes are then absolutely mechanistically required for the differentiation of Treg progenitors into inducible Tregs. Now, I'm just going to close on this line of discussion with the observation that 2 other themes that pop up here. Very consistently, we get changes in transcripts related to stress granules and the immune synapse. Well, let's change gears. So I was giving a talk kind of similar to that at Grand Rounds at GW. And after the talk, one of our intensive care physicians, McCaffrey, came up and said, "Hey, Tim, I get this. RNA is the future. RNA-based diagnostics are slick. But if you can diagnose chest pain, then you really got to help us out, and you really got to help us diagnose abdominal pain." And, you know, I'm kind of stupid about things like that. And I thought that diagnosing abdominal pain was already pretty much worked out. And he points out, well, yeah, it is in maybe 30 or 40% of cases, somebody presenting with lower right quadrant pain, kind of shown over here. rebound pain at that position, unable to lift their right leg. Yeah, there's some signs that we're very confident. You send them for a CT and you're very confident that they have appendicitis. But then there's another 50% where that is not at all obvious. And so we did a pilot study in which we ran the protocol that I'm talking about before. We took blood from these patients presenting at the GW emergency room with abdominal pain. And then as controls, we used people that had just something like a hernia, just a purely traumatic cause for their presentation. And we thought they were going to be controls because it was in the lungs, not the abdomen. We took 5 people with pneumonia. In this case, we ran them on Illumina bead chip arrays, but took stabilized blood. And then we compared those transcript profiles to the outcomes. And I'm going to simplify this for you. If we looked at transcripts that were reduced in the patients with appendicitis, there was a cluster of transcripts that are related cassette genes called defensins that went down very strikingly in green here in the people with appendicitis. But then in people that had appendicitis or lower respiratory infections, there were 2 transcripts in particular, IL-8RB, which is CXCR2 for those of you that are chemokine groupies, and ALPL, which is alkaline phosphatase. You can see that the people with lower respiratory infections or appendicitis were generating 6- to 7-fold increases in these transcripts. And so we thought, maybe we have something here. And one of the first things you want to do out of array or RNA sequencing studies is try and validate it by a different method. Show that a different measurement method will give you a roughly similar answer. I'm going to just very briefly give you an endorsement for what's called droplet digital PCR. There's other digital PCR platforms, but they are honestly stunning. You take your RNA and you parse it into individual nanoliter droplets, and then that is cycled so that each of those becomes a chamber, if you will. Those chambers are then either positive, they show a positive PCR reaction, or they don't. And then you use the number of positive to negatives against, this is Bayes' law, to actually quantitate the number of copies of that transcript. And you don't need a standard curve and you don't need duplicates. And we get amazing reproducibility and accuracy from it. Make a long story short, even by PCR, we could readily confirm these in a completely new set of patients using ddPCR. We could confirm that in, for instance, people with abdominal pain, they had elevated defensins, they had elevated IL-8RB, and then the combination marker, which, as I'll tell you, we think is a biofilm marker. And in the people with lung infections, it was really almost exclusively the defensin marker that would be elevated in those patients. And this is how we think it works. And what you need to know is that when we started this back in 2016, 2017, you could not get physicians to give you a straight answer to whether appendicitis was an infectious disease or not. And yet since then, it is now, for instance, in Europe, 80% of cases of appendicitis are treated with antibiotics. It is now accepted that appendicitis is almost certainly a biofilm infection of the appendix. It can be caused by blockages or physical effects of accessibility of the appendix, but that biofilm infection causes inflammation of the surrounding tissue and secreted factors that then go out and activate the neutrophils. We pick up these neutrophils out in circulating blood, and then we're detecting changes, particularly in what I told you, ALPL and IL-8RB are our measures of biofilm. When we have more time, we'll talk about the many wonders of biofilms and why they're probably a really interesting diagnostic target in a lot of areas of medicine. Well, so then you all know what happened. COVID broke out. We were in the middle of a multi-center validation trial on the coronary markers, and then they shut down cath labs. Thank you very much. So we had to pivot, as was alluded to, and we did. And many thanks to the emergency room physicians and the intensive care docs that helped us grab these patients in the middle of a pandemic. And these are now patients where we tried to determine whether our infection markers would also help us diagnose the host response to COVID. And we got a total of about 48 patients that went into that we could get blood from the day that they entered the intensive care unit. They were then classified as being incidental. So they were SARS-CoV positive in a nasal swab, but that was not their cause for admission. That's an important point. People were still going to the ICU for pulmonary embolisms, for car accidents, gunshot wounds. You know what gets you into the emergency room. There was another group of people who were positive for SARS and had symptoms, but it was not severe, as in they were not intubated or did not need vasopressors. And then, as you probably are aware, in severe COVID-19 cases, they were frequently intubated. They were put on vasopressors to maintain their blood pressure because they're getting shocky. And then a criteria also that got you in this category is that you ended up dying of COVID-related symptoms. Now, where we're going to adapt this just a little bit is in the interim, we have learned to isolate, and this was about a year's worth of work of a bunch of ways that don't work very well, include sucrose gradients and all kinds of other ways to isolate neutrophils. What works very well is actually magnetic isolation using CD15 coated magnetic beads. And I'll show you a method we developed that we believe is almost ready for point of care diagnostic of neutrophil activation. But to continue our discovery program, we took some of that blood and we put it on Illumina total RNA sequencing at our genomics core to confirm these markers. And then we compared it with lab values over there on the right. And blissfully, I will admit, some of the targets that we identified by COVID-19 were similar to ones that we had identified in abdominal pain and in lung infections. So the transcripts that were going up, I'm just going to highlight a few of them here. ALPL was going up and the defensins were going up, and another neutrophil marker, 16-fold increase in amyloperoxidase, a known neutrophil marker, 20-fold in defensin. But again, put this in your pocket. Cathepsin G goes up 13-fold. Only some of them are shown here. There's 600 that go down in the patients with COVID. They are almost all T-cell related. So when you take these transcripts that go down and you put them into so-called gene ontology analysis, the signal is overwhelming, as in E-37, that these transcripts are related to T-cell exhaustion. Okay, so using the analysis that I showed you before of what cell types are involved, the patients with COVID had strongly increased transcripts that related to neutrophils and decreased transcripts related to T cells. B cell transcripts increased, and these are the eosinophil transcripts, less than the neutrophils. But the point is, clearly the T cell transcripts are going down and the neutrophil transcripts are going up. Well, in relationship to the severity of the disease, we got much the same thing. If you broke the patients by who needed vasopressors, so you found pretty much the same thing. You find that the defensin markers are going up 5-fold, and this is just emblematic of all the T cell markers. They're going down more than 2-fold. In patients where the COVID was fatal or not fatal, pretty much the same story. The defensins are increased, the neutrophil markers going up, and the T cell markers going down. So this very consistent trend that is also consistent with the published literature, that the severity of COVID is determined by the relative decrease in T cells and the increase in the neutrophils. And this ratio of sort of T to neutrophils, we think is sort of correlates or may even be causative of the so-called cytokine storm. So put another way, the degree of disease is not so much determined by the pathogen itself. It is at least partially determined by the severity of the reaction to that pathogen. Now, let me just give you some idea of how well these markers work. So here's the clinical lab values on these patients. These are historical values for white blood cell counts, neutrophil count, et cetera. And in the various groups, then these are our incidental, moderate, and critical. And you can see something like a white blood cell count is not terribly helpful for in diagnosing the severity of COVID. Other conventional markers like that are routinely used that are inflammatory markers like lactate, CRP, creatinine, really, really not helpful. By comparison, our RNA markers are really extremely helpful, wildly significant over baseline levels of controls, with in particular ALPL being a very useful marker of severity, defensin also a useful marker of severity. And so we can now combine these into a RNA positive score, and it works really, really well. It's about probably 95% accurate in predicting disease severity. It's predicting really the host response. Let me make a quick point that surprised me a lot. One of the things that we did right here is we took the blood and we correlated, we took the blood, measured the titers of SARS-CoV-2 by ddPCR in blood. So we could determine the levels of RNAemia in these patients. And to our surprise, the people that were critical COVID versus people that had incidental COVID really had not striking differences in the blood levels of the SARS-CoV-2 virus. In each of the groups, you had a few patients that had very high titers of virus, but the majority had either low titers or full-on no detectable virus in blood. Now, they could and did have detectable virus in a nasal swab, but at least in terms of their circulating virus, there was really no relationship between circulating virus levels and the severity of the disease. Now, said another way, in the lower panel is the correlation between the RNA markers, or let's take DEFA or ALPL, and markers of clinical severity like SOFA and the Apache score. And you see highlighted here the number of actin B copies, the DEFA levels, the MPO levels are positively correlated with the SOFA score, which is really the number of organs failing in the patient, the severity of the disease. But by comparison, the titer, the viral titer, has almost no correlation to the severity of disease or to our RNA markers. So the virus itself is probably more like a trigger to an inflammatory immune syndrome, much like septic shock, if you will. And the virus itself is not really what our RNAs are tracking, and it is not really a major determinant of the severity of the disease. Okay, let me wind up actually really quickly now on what we discovered was that as we started looking at these cells that we were isolating, we noticed that there were many more neutrophils, obviously in the people with COVID-19, but that the nuclei were much brighter. And to cut to the chase on this, we've tracked this down to the liberation of elastase from the granules as a way going in and cleaving the chromatin and making it more accessible to the fluorescent dye. And this is just a series then of the COVID patients, and we developed an assay for their elastase activity. You can see their elastase activity is much higher than controls over time longitudinally. And what's kind of interesting is that these cells are viable. And if you look at the end of, say, 30 minutes here, the neutrophils that we've isolated are now starting to form nets. And those of you that have been following COVID know that the netosis, the release of these nets, neutrophil extracellular traps, is thought to be one of the really punishing aspects of the disease because it clogs the airways and it clogs vessels. In the interest of time, I'm going to skip this. I'm just going to say that what the CD15 count is, of course, higher in people with COVID, and that you get a bonus effect. The elastase activity, even on a per cell basis, is elevated in the patients with COVID. This just shows that actually you can show that in the cells, but if you try and purify and measure the elastase in plasma, it doesn't work. And I think that that's why this has not been used clinically to date. Okay, so I'm going to close with a study on abdominal pain, and I'm going to do it very quickly because if you're interested, we are starting now a multi-center trial of using these RNA markers to diagnose abdominal pain. In all cases, they go through a standard workup, they get a CT scan. What we did is we've run the neutrophil assays that I showed you. We think we might have this to a point where it's ready for point of care. But this blood RNA assay where we quantify by ddPCR, we have a lot of confidence in. Let me skip this. It just shows you that in the abdominal pain patients, these transcripts are significantly higher. That's not what matters. This is the impressive part right here. When you take these patients, there was a total of 427 patients were screened, 100 were enrolled. We got quantitations on 89 patients with a firm diagnosis. 10 of them ended up having acute appendicitis. Our RNA markers are positive in 100%. But if you look at a white blood cell count, if you look at the neutrophil lymphocyte ratio, or you look at a lactate score, you don't get better than 50%. Roughly the same with diverticulitis, cystitis, pyelonephritis, which are all rather known to be caused by infectious agents. Other disorders are less clear about whether infection is a root cause. Things like bowel obstruction, things like obstructive renal stones can have an infectious etiology. And in fact, in 80% of those patients, we pick up an infectious signal, and yet by other means, you do not. And then if you go down into the really what I think everybody would agree are non-infectious causes, something like a ruptured ovarian cyst, we get only one of those patients that has a positive RNA signal. And non-obstructive renal stones, we get none. Where I think the real value here is right here. Of these 89 patients, 18 of them are discharged with no organic cause for their abdominal pain. But in 50% of them, we get a signal that tells us they have some type of abdominal infection. And you would not really pick that up just looking from their white blood cell count. So we think this is a great assay. I'm not going to go into the detailed statistics here, but if you want, you can get the ROCs and things like that. I'm going to focus on the bottom line here, which is that in the patients from the emergency room that were admitted for a cause potentially relevant to an infectious disorder, 33 of 34 cases, 97% of those patients had a positive RNA score. We think this is a very, very sensitive test for abdominal infections, and we very much want to regulate that and validate that in a larger trial. In the interest of time, I am going to just tell you that we think we have developed a point of care test shown here, where a small amount of blood can be put into a 3D printed chamber. The magnetic beads isolate the cells, wash away everything irrelevant, and then we measure the elastase activity, and it actually only takes less than 15 minutes for that part of it. So all told, we're talking about a 30-minute test that's probably $10 a piece. We're pretty excited about it. We'd very much like to collaborate with the DC hospitals and medical centers to help validate that. This is the trial. If you guys are interested, please reach out to me after the talk and we'll talk about it. Got to take the last minute to thank everybody. I am blessed with really wonderful collaborators in my lab, and a particular shout out to both Ian Toma and Richard Wachowski, who've done a lot of the heavy lifting on this. Our emergency room physicians and our ICU docs get an extra bonus because when you think about it, they recruited all of those patients for us in the midst of a pandemic. So bless them. I'm going to leave it there, and again, extending the opportunity to collaborate wherever this works out for you.
Okay. Thank you, Dr. McCaffrey, for that exciting research. There's just so many elements, right? We've got looking into the like cellular physiology, what's going on at the molecular level, as well as the technology, right? And how we can take this from the bench to the bedside. We're going to stick with COVID. There's a few questions. Did you check on the strain that the patients had? And do you think that would have made any difference in the levels of the transcripts?
So we did not do a particular sequencing at the time. This was all done, those were all recruited in the midst of the Delta wave. So we feel pretty strongly that they were very likely Delta positive. That said, we have been re-recruiting. We reopened during Omicron and got another 20 patients, and we pick up a strong signal in the Omicrons as well. Probably a point I didn't have a chance to make is, regardless of whether it's a virus or a bacteria, our strategy is agnostic. So it doesn't really care what the pathogen is. All it cares is that your immune system is activated.
Got it. And do you intend to follow long-term these COVID patients at all? Is there any interest in longitudinal studies?
There is interest in it. We very much like to do that. I think there's an open question. I think that the long-term COVIDs, I think it is reasonably accepted that they do not have ongoing viral infection, but I think it is not known whether they could have unrelated, for instance, biofilm infections or bacterial infections that contribute to their long-term disability. So our test actually would be quite amenable to that.
And this question about, is globally, do we think that, do you plan to have collaborations with other countries and other populations to see if we're seeing the same level of transcript changes that we're seeing here?
Right. We think so. And that's sort of the beauty of why I was explaining the difference between DNA and RNA. So DNA can be, to some degree, ethnically and geographically separated and polymorphic. But RNA expression, particularly in your immune system, really should not. Someone in India should show the same upregulation of defensins as someone in Japan or America. So yes, it's absolutely something that we want to test, if for the main reason that these blood-based tests are going to be vastly more amenable to lower resources countries than something like a CT angiogram or MRI for abdominal pain. So these are very low cost tests that are very, very amenable to diverse sort of working environments.
So with that, I'll ask the question, what are the validation steps do you think that this biomarker should go through to be used for clinical diagnoses?
We've on the coronary, we've been to the FDA, so we know what steps they want, and it's a little bit surprising. They are not interested in massive numbers. They are interested in a representational cohort. They're interested that you've tested your markers in the same population that you intend to use it. They are mainly, quite reasonably, preoccupied with whether you can measure the same person the same way on 2 different days. In other words, the reproducibility of your measurement. Said another way, how much error is in your system. So they're very interested in technical features of it. And so we have a study designed, for instance, on the abdominal pain markers that is in the range of 500 to 1,000 subjects, and it does need to be multi-center. But unlike therapeutics, diagnostics are much more sort of FDA friendly.
Right. And we've got, let's do one more question here before we turn it over. So this is a perfect example of your bench to bedside, right? So how long do you anticipate working on this in the laboratory before it could be deployed in patients?
That's a good question. I think that if all goes well, I think the RNA-based tests, we have a great deal of confidence in it. I think we could be through validation trials and in front of the FDA within a year.
Good news. Good news. Okay, well, good luck. And yes, if there's other those in the audience who are interested and have follow-up questions for Dr. McCaffrey, please either contact the hosts of the conference or email Dr. McCaffrey directly. So we're going to keep an eye out on that technology and your research, and hope for the very best. And this is, like Dr. Pam Norris mentioned, this is one of the more positive side effects, right, of the pandemic. And so thank you again for your keynote address. And with that, I will turn it over to Dr. Jane Otato for panel 1, long-term COVID-19 impacts on the body. Thank you.
Okay, that was a really good presentation, really good, interesting clinical trial studies. So the next we have Dr. Ashcroft, and we have Dr. Rim and Dr. Sen. So I'll start introducing each one, and then we open up the panel. Dr. Ashcroft received his undergrad degree in medical technology from Shira University, Iran, master's degree and PhD in molecular biology, Utah State University, and has 2 postdoc training, one in Indiana University and the other University of Florida. Actually became the first holder of Fulbright scholarship from Howard University, and his country a Fulbright ambassador in the Washington, D.C. metropolitan area, and also serves on the board of directors for the SNI Health Foundation of North America, a nonprofit organization that provides help to uninsured people in the D.C. metropolitan area. He's a professor in the departments of medicine and genetics and human genetics at Howard University, served as a member of gastrointestinal working group in Howard University Cancer Center. And he's the director of microarray facility at Howard University. And he has more than 175 peer-reviewed articles and been invited to many GI special conferences as keynote speaker. He's a member of several NIH studies sections since 2005. His group focuses on epigenetics, genetics, microbiome, and epidemiology of gastrointestinal cancer, especially colorectal cancer. His current research has identified novel mutations in African Americans with colorectal cancer, which were highlighted in the Journal of Cancer. His group is working towards biomarkers of identification in early to late colorectal cancer. Dr. Brem received his PhD in the field of environmental microbiology from Free University in Brussels in Belgium. He then moved to the United States, where he continued his field at the Uniformed Services University of the Health Sciences as a research associate in many original and high-profile publications in Nature Biotechnology, Applied Environmental Microbiology, and Microbiology. He has more than 100 publications in microbiology and microbiology. He joined Howard University. He's currently an associate professor of pathology, and his main research focuses in gastrointestinal oncology in minority population. One of his primary interests in this area is the role that the gut microbiome plays in oncogenic transformation. He's a holder of different grants and member of the editorial board of several journals. Dr. Bream is presently using his expertise in gut microbiomes in patients with sickle cell disease to determine specific and features of their gut microbiome. He's also analyzing a gut microbiome dynamics in obese and lean subjects under different diet regimens. In the pandemic era, he's extended his expertise to analyze the potential role that the gut microbiome highly play in the long COVID. Dr. Sabaiti Sen, I hope I pronounced Wainwright, forgive me if I didn't pronounce it right. Is Associate Chief in Epidemiology, Veterans Affairs Medical Center, Washington, D.C., and he's also a professor in medicine and biochemistry and molecular medicine at George Washington University School of Medicine and Health Sciences. He's a board certified endocrinology and internal medicine. His transnational research interest involves studies on how high glucose affects the stem and progenitor cells. He has studied diabetic and metabolic syndrome using adult stem cells, not only in the regenerative field, but also has used them to predict cardiometabolic health status in response to an intervention. He has received funding from American Heart, American Diabetic Association, and NIH. The goal of his current research is to determine the long-term effect of COVID-19 on cardiorenal and metabolic health in the scenario ongoing on pandemic. I welcome the panel, and the floor is yours right now.
Good afternoon. Am I going to start?
You could start.
Okay. I just asked for sharing authorization. If you can give me share, I can start. Okay. Now I got it. Thank you. Okay. Everybody see my slide?
Yes.
Oh, okay. Good afternoon, and thank you for inviting us to this very important, significant panel. I think it is the right time to discuss the heart of COVID pandemic, which is really bothersome, and that is the part of the race, which is very important in terms of the infection, death, hospitalization. So in this, I am at the GI division at Department of Medicine, Howard University. My interest is mostly genomic and colorectal cancer, but due to the pandemic, I had to switch a little bit the course and move to COVID because the lab was almost closed, and then we had to do some arrangement, and so we went through the COVID patient, which we have seen with a lot of GI manifestation and symptoms. And so going through that, I thought that bring you just the latest science news, which was published just recently. And this is building out what we have learned in last 2 years, which is a lot we learned. And one of the learning process is Zoom, which we talk right now on the Zoom, not in face-to-face. And hopefully we can make it face-to-face because the innovation based on nature, which published yesterday, is more when we see each other and make a network. Therefore, I think what we see here is COVID after 2 years, we have learned a lot and we need to learn more. And these 2 years, a lot of questions raised. And this question, you can see that is evolved from January 2020 to almost January 2022, when we didn't know what the virus is, how is caused pandemic, what is the effort of non-pharmaceutic intervention, and how it's dominated this transmission, and what is the driving the waves of infection? Can reinfection occur? And is the virus adapting? As we go forward, we see that coming to the vaccines and then the effectiveness of the vaccine, and how is the SARS-2 containing the adapt, and is it evolving to escape immunity? I said Dr. McCaffrey talked about some of the immunity, neutrophils, and the abdominal pain, which is part of really the first or mostly one of the first symptoms in GI manifestation and COVID, abdominal pain. And what you can see in the word, this is the black curve. You can see this is the confirmed infection in the ward, which is up to January 22. And then next you can see in Europe, then you see North America, then Asia, South America, you can see Oceania, and then Africa. So Africa is really level, as you can see. And this is part which I thought it is very important we all think about, like Nigeria. Nigeria, less than 4,000, officially less than 4,000 deaths, but more than almost 150 million people. And Ethiopia, so we had communication with the people there to see what is going on, but this is the official death report which has been seen. There is something really going on there which we have to understand. Now, this is Africa. How about the race in America? So you can see this is the latest CDC report, which came and talked about the case, hospitalization and death. And this is comparing to rate ratio compared to white non-Hispanic person. You can see American Indians has 1.6 more than the whites for in the cases. 0.7 is Asian, Black, or African American, 1.1 for the cases. Hispanic is 1.5. Hospitalization, you can see in American Indians, 3.1, almost more than threefold more than whites. Asian in 0.8. African American is 2.4, Hispanic 2.3. And the death is in American Indians is 2.1, Asian 0.8, African American is 1.7, and Hispanic is 1.8. So race actually play a role here. And why is that? And what we can do about it? We thought that this is something which we can talk about, the GI related issue, which is something which we can perhaps talk about. And this is the code among African American and Hispanic. And we said, how does GI symptoms impact the outcome? So we had at Howard University at the time, which we had about more than almost 500 patients. So we know that coronavirus disproportionately affect African American and Hispanic. We know because they are workforce and they get exposed, and then socioeconomic status, access, and the infection rate is high in that group. We retrospectively evaluate the medical records of 386 COVID positive patients admitted to Howard University Hospital between March and May 2020. We assessed the symptom, including GI manifestation, comorbidity, mortality, using logistic regression analysis. By the way, our patient population is at Howard hospitalized is more than 1,000 right now. This is what we have had for this study. And then what we have seen that out of 386 COVID patient, 257, which is 63% were African American, 102, which is 25% Hispanic, and 26, which is 6.4 is white. And then the mean age was 55 years old. However, the mean age for Hispanic was lower, 43 years old, and for whites was 61, and for African American 60. Mortality rate was highest among African American and lowest among Hispanic in this study, of course. Patient with shortness of breath and elevated aspartate aminotransferase, AST, elevated procalcitonin, ferritin, and lymphopenia had high mortality rate. Cough and fever were common but unrelated to outcome, and hypertension, diabetes mellitus were the most common comorbidities. And glucocorticoid treatment was associated with high mortality, and this is adjusted for age and race. Diarrhea was prevalent, and the GI symptom did not affect the outcome here. Liver enzyme alteration is associated with death in minority COVID-19 patient. As we see, AST, ALT is elevated in the COVID patients. Now, in our conclusion, we have African American, our study had the highest mortality as they considered an older population and have comorbidities. Age is the most important factor along shortness of breath in determining the mortality rate. Overall elevated liver enzyme, ferritin, procalcitonin, and C-reactive protein were associated with poor prognostics. Diarrhea was the most prevalent GI symptom. However, GI symptoms did not affect outcome. Glucocorticoids should be used judiciously, considering the poor outcome associated with it. Attention should be given to monitor liver function during COVID-19, especially in African American and Hispanic patients with higher disease severity. So I thank you. I am happy to answer question at the time which moderator would like to ask.
So Dr. Brahim would want to go next?
Yes,
Sure.
please. Good.
Yeah, you can.
Okay, thank you, Dr. Hurtado. So basically, I'm gonna just have 3 slides, and I will go through the material I want to present here. So I'm working in the Department of Pathology at Howard University, where along with Dr. Ashtrab, we are working primarily on gastrointestinal oncology. And just to give you a background of why, in addition to the fact that the labs were closed, why we have this interest in COVID, we published a paper within the pandemic, even though the work was done before and was submitted before the pandemic, on HPV-16 lineages and anal cancer in African Americans. And the relevance of that paper as far as the pandemic is concerned is that we found that one HPV-16 sublineage, what we might be referring to these days with SARS-CoV-2 as the variants. So one sublineage of HPV-16 that is not oncogenic within whites is highly oncogenic within African Americans. So the reason I'm bringing this up is just to show that viruses or sublineages or variants of a given virus might show different outcomes in different hosts. As a matter of fact, in the early wave of the pandemic, the Alpha wave, Hispanics had a good high incidence, but still they had a mild outcome or good outcome with the pandemic, at least in the first wave. But that changed at the time of the Delta wave. In the Delta wave, Hispanics also start showing severity and start showing deaths similarly to other groups. And if you all noticed with the Omicron wave, it leveled the ground for all, both nationally here in the US, as well as globally, since there were very few deaths and very few severe cases reported worldwide and also here within the US. So that brings us to the fact that different variants associate with different outcomes and also with different outcomes in different populations and different ethnicities. Since this panel is about long COVID, the data that Dr. Astro was showing We are still working on that. We are still following up with several of those patients right now, trying to collect their outcome months later after they were discharged from the hospital. And we are putting those data together right now just to try to make sense of it and see which people have or which patients do have primarily persistent or recurring or new symptoms. The most important publication so far in the context of long COVID came from the University of Seattle in a Cell paper where basically they showed that diabetes, SARS-CoV-2 detection in blood, Epstein-Barr virus, as well as autoantibodies, these are 4 major risk factors for long COVID. So diabetes is one of the most important one, while it is not related to poor outcome during hospitalization. It seems to be highly impacting post-discharge symptoms. Epstein-Barr virus, there is a major steps of reactivation of viruses. For now, only Epstein-Barr virus is cited, but most likely other viruses as well will be cited in the near future. And then we have patients who are showing autoantibodies where basically autoimmune disease are starting to show up. And I agree with Dr. McCaffrey when he was saying that it's not really the pathogen itself that will be driving the disease later on, but more the inflammatory response. And this is the case also for long COVID, where we have these autoimmune antibodies are showing up in patients who have long COVID. If we go to the next slide. So as I stated, this was a paper that was published in Science by one scientist from UK, Dr. Alwan, where she basically showed in one very comprehensive slide or figure what needs to be done to capture, monitor, and then later on potentially treat long COVID. If you go to the next slide.
We are there.
Okay, so basically there are 3 major steps in the context of long COVID that needs to be fulfilled here. And as she summed them up there, 1 is report, 2 is recognize, and 3 is research. So as far as reporting, it is very important for all of us to not dismiss patients who come up with symptoms post-discharge. And I have seen this personally in our hospital where patients, for example, at least 2 cases that I know personally, where 2 patients had persistent breathing issues and all kinds of batteries of tests, cardiopulmonary were done on them. And they were said that basically you don't have any tissue damage. Everything looks fine. So this is just has been dismissed as some kind of psychosomatic disease, not a real physiological one. So reporting is a major thing, and we're going to go to the next one, recognizing in a minute. So reporting means that we have to sensitize our patients to be alert about subtle changes in their overall health. And just if we do not do that, if we do not sensitize our people, our patients, especially in our case, our minority patients, this on its own will skew the overall picture that we're going to see, or the landscape picture of what is long COVID exactly and who it is affecting. For example, we know that men are less likely to go to the doctor because of minimal or minor symptoms. We know as well that minorities are very likely not to show up to the doctor for minor symptoms as well. And just these 2 facts will basically change the real landscape or the real prevalences of long COVID if we do not sensitize our patients to be alert and to report anything and everything they experience post-discharge. It is also important not to put into the long COVID anything that shows up post-discharge, because in many cases also there are some artifacts that will show up, especially in patients who have gone through mechanical ventilation, which is on its own associated with long-term months associated symptoms. Or in some cases also, these symptoms might be really not real, and we can dismiss them, but after we dig deeper into those. And that brings me to the second part of this slide here, which is basically recognizing, and this is where the role of the clinical staff comes in. So no need to dismiss. It is good to explore and to collect the information and then follow it up with whatever can be objectively measured. And this is where blood tests are important. For example, in the context of our NIH Recover project that we are part of the consortium for NIH, we are following hundreds of patients longitudinally for 4 years, and we are collecting all kinds of data. We take whatever the patients are reporting, but at the same time, we try to see if we can validate any of those symptoms that are reported by metrics such as blood tests, and trying to see CRPs, troponin, procalcitonin, D-dimers, ferritin, and all those kinds of markers that would, in principle, allow us to validate some of those symptoms. So recognizing is basically collecting all those information, not dismissing what the patients are reporting, and then using objective metrics to basically try to correlate what the patients are reporting with the symptoms that they are showing. And the third major component here is researching, and we have a good example in Dr. McCaffrey's presentation. And basically researching here is the goal, for example, for the NIH Recover study that we are part of, are three. One is to establish the real prevalence and incidence of long COVID. If you look at the publications right now, it goes from 5% all the way to 70%. So that means that it's a wide range that needs really to be studied further to see the exact percentages we are talking about. You remember that officially right now we have 80 million reported COVID cases in the US. And this is an underestimation of the real numbers since we have been testing home recently. And it's very likely that we have something like at least double that number, 160 million people who have had COVID. If we are putting the 5% to 70%, we are talking about a major size of the population that might be affected by long COVID. And this is why it is very important to, one, capture this long COVID, two, define what long COVID is, and see whether there is one or several phenotypes of long COVID, and then research what are the risk factors that are associated with long COVID. And in that context, one of our proposals is that we think that the gut microbiome is one of the major elements that is playing a role in long COVID. Many of the patients with the COVID-19, they end up having this dysbiosis, which is basically a narrowing of the diversity of their gut microbiome. And that in itself has major systemic effects, both for inflammation, as well as for blood coagulation and thrombosis, as well as for microclots in the system. So this is why we are suggesting that, and we are pursuing that in the context of our studies. Worth noting that in 2017, there was a publication that came out referring to 1 century after the Spanish flu, and they defined something called encephalitis lethargica, which was the name that they gave to the post-Spanish flu pandemic of that time. So when you look at the definition of encephalitis lethargica that they defined there, and you look also at what is referred to recently as MECFS, which is myalgic encephalitis chronic fatigue syndrome, many of the people in these areas, they say that you guys looking at long COVID, you are just looking at the same thing, you are reinventing the wheel. Most likely you are just looking at the CFS, chronic fatigue syndrome. And that remains to be seen. And this is why this longitudinal study that we are part of and so funded by NIH will answer those questions. We will find really whether there are some specific patterns, phenotypes, and subphenotypes of long COVID, and how we can assess the risk factors, and then define some specific clinical trials to address long COVID in our populations in general, and in our subpopulations and ethnic minorities as well. Thank you.
Okay, thank you, Dr. Brahim. Dr. Sabayachi Johnson.
Hi there.
Welcome.
Thank you. Thanks, Dr. Brim and Dr. Ashraf. Very interesting talks. And of course, Dr. McCaffrey, who is actually my colleague in crime to a certain extent. Thanks, Tim. And thanks to the entire team who has worked tirelessly, Dr. Sanders, Priscilla, and everybody else who had helped us to get this up and running. Anyway, let me try to share my screen. I think somebody else is sharing. Oh, hold on. Or maybe. There you go. Can you guys see my slides?
Yes.
Awesome.
Yes, we can.
Okay. So I'm an endocrinologist. I'm also board certified in lipidology and obesity medicine, and of course, internal medicine. I have been in GW from after coming from Tufts since 2013. And my prime interest is cardiometabolic diseases. And as you just heard from Dr. Brim, that patients who has diabetes or even pre-diabetes stand to have the biggest impact of this COVID. And I don't think it depends on what's the serotype, what's the particular genomics of the virus that we're talking about, but it clearly causes long-term damage. And I'm going to talk to you about kidney damage or podocyte damage. And we actually have some data on it. So let me see. Okay. So COVID, as you know, COVID-19 is a symptom complex, which is attributed to the virus infection. As you heard from the previous talks, that it is associated with endothelial cell dysfunction, which is manifested by clots and vaso-occlusive diseases. And it's hallmark of type 2 diabetes with renal podocyte dysfunction is often an early manifestation. Now, I'm going to talk later on about exosomes also as a means of understanding what is the secretome from different cells in order to understand what cells are getting damaged in this process. And we looked at specifically exosomes coming from renal podocytes, which, as you can imagine, is mostly in urine. So we looked at urine exosomes to specifically study kidney damage. And I'm going to talk to you about why suddenly we decided to look at kidneys. So in this study, we also explored how does hyperglycemia predispose and increase SARS-CoV-2 and its specific relevance to the kidneys and endothelium per se, right? So our study design was approved by the GW Medical Ethics Committee, and we looked at SARS-CoV patient samples from blood and urine after resolution of their symptoms at week 10, which is approximately 2.5 months, 6 months, and 12 months from the acute phase of infection. We collected mononuclear cells from the whole blood by Ficol separation and centrifugation. We also looked at CD34 cells, though I'm not going to talk about it today. We specifically looked at from urine the exosomes, which we obtained by ultracentrifugation of the urine. And we did gene analysis studies and exosome-based studies that I'm going to talk about now. So the reason why we decided to look at kidneys when everybody else was looking at heart, lungs, and so on, is we exposed multiple human cell types to hyperglycemia. And we looked at primarily the expression of the receptors that the virus uses to enter the cells, such as ACE2 and TMPRSS2. And when we looked at the human kidney cells grown in presence of normal glycemia and hyperglycemia, as you can see, the gene expression of the receptors that the virus uses in these kidney cells was significantly higher than compared to normal glucose. So once noting that, we said that, you know what, maybe we are actually missing the damage that the virus is causing to the kidneys. And simply because we are probably not looking at it. We looked at the blood glucose of these patients at various time points. And as you can see, these were, though random, these were patients who were 10 weeks out of acute symptom, right? 6 months acute symptom. These patients were not diagnosed to have diabetes, okay? But as you can see, their random blood glucose is clearly high. That means they might have developed endothelial dysfunction leading to associated insulin resistance and so on. So, but the point of the fact is they are essentially in hyperglycemic range. When we compared their colony formation from the mononuclear cells, and colony formation is essentially the cells coming together to try to form an organelle. And as you can see, these numbers were actually lower, almost similar to a patient who has type 2 diabetes for almost 5 years, right? And clearly these samples are easily obtainable by me, but then I was surprised to see that their colony counts was almost as low as patients who have bona fide diabetes compared to patients who did not have the diagnosis. So you can see that the imprint is not just about hyperglycemia in the serum. The impact has gone way beyond that and has been implemented in the lack of colony formation in these patients who are almost 6 months out of acute symptom. And now coming to the so-called exosomes, and let me explain why podocyte-specific exosomes. The podocytes are essentially, in some literature, would be classified as modified form of endothelium. They are essentially a barrier between the urinary structures and essentially blood and urine. It's almost like endothelium is the barrier, the last barrier from your blood to the other organs, right? And it actually has a lot of secretomes. It produces a lot of proteins such as nephrin, podocalcin, Wilms tumor. As you know, there are certain cancers that we detect looking at Wilms tumor protein. So it has a lot of factors which keeps the renal glomerulus safe and functionally well. So if anything is damaged, such as podocyte inflammation, which is known to be hallmark of certain cardiometabolic diseases such as type 2 diabetes. Type 2 diabetes with histological sections have again and again showed podocyte inflammation, right? But the worst part is for any kidney disease, and I've worked with my nephrology colleagues in GW at VA, there are essentially only 2 biomarkers, whereas kidneys, brain, liver has multiple of them. And as you heard from Dr. McCaffrey, when you talk about heart, there's no lack of choices of biomarkers. For kidney damage, which from diabetes is affecting the minorities in massive numbers, and any non-Caucasian population, particularly if you walk into a dialysis unit in GW or anywhere in the city, you would see mostly non-Caucasians, and it's very under-investigated. Now, if you look at the biomarkers for kidney, there are essentially 2. EGFR, which is estimated, and 2 is proteinuria. That's it. Unless you do a histology of kidneys, you just cannot diagnose a progressive kidney disease. So we essentially wanted to look at podocyte-specific exosomes so that we understand and realize what is the first steps of kidney damage. And we essentially use other biomarkers of exosomes as our quantification to see the protein amount. Let's go to some of the data. Here is the nephrin 2CD9, which is a biomarker, is a standard biomarker, a ratio, and you can see that at week 10, 6 months, 12 months out of acute symptom, the levels are almost 2 times higher than a type 2 diabetes patient's urine sample. Now, there are multiple patients in this one. We have about 16 patients here. We have about 8 here and much less here. And we are actually continuing to do this study. This was purely GW based. We are continuing to do this study in veterans, which I'm not going to show here, but we are at this point getting more patients here. But as you can see, there is a statistical significant increase even when you compare the podocyte specific proteins, even from an established type 2 diabetes patient. Now, this could be the fact that these patients are just about getting the podocyte damage. These patients have already established podocyte damage. So that may be the reason of difference, but clearly there is a significant statistical difference between the protein increase, and this is persistent. The range of protein, podocyte specific protein excretion in the exosomes has been persistent even up to 6 months. Now, this is not just one protein. We have also looked at podocalixin, which is almost the same or similar to what we found in nephrin when compared to type 2 diabetes patients. So we believe that this is actually a very alarming situation. As you know, to prevent a patient from getting progressive kidney damage, we have very little medications at hand. We cannot control their blood pressure, their proteinuria by giving ACE inhibitors. If we know that this is going to happen, maybe we should institute that sooner than later in long-COVID patients. So in conclusion, the presence of hyperglycemia clearly augments the receptors that the virus uses to enter a cell, particularly the kidney cells. And it's not just about kidney cells. They do so in lungs, endothelium, and so on, but the kidneys are not very focused on, unfortunately. But diabetic kidney disease is the largest complication and rising amount of patients in this geographical area. Podocyte-specific urinary exosomal protein estimation seems to be more specific than urinary albumin. The reason I say that is you don't see a huge lot of change in urine albumin protein estimation, but when you look at the podocytes, you see a significant statistical difference. And clearly the urine protein, when we estimate it, it's clearly not a cell specific. So whereas our estimation of exosomal protein is podocyte specific, which is how the diabetic kidney disease starts. A persistent level of podocyte-specific protein was noted even at 1 year post-COVID. We are now actually focusing at 1 year and 1.5 years post-COVID to see whether these patients are truly continuing to develop renal damage as featured by common clinical presentations like hypertension, increasing proteinuria, and so on, which would cause a huge problem in years to come. So thank you for listening to me, and this is my team. I do collaborate with Dr. Bukrinsky from Microbiology in GW and his colleagues, Dr. Brichacek, and my lab manager and senior scientist, Dr. Nandula, who did a lot of this work. So thank you.
Thank you so much, Dr. Stan. And it's a wonderful presentation. And now welcome all the panels in, and any questions that you might have, please send it to the Q&A. We got one question here for Dr. Brum. Dr. Brum, is there a recommendation schedule and testing panel for post-COVID patients who do not present any remarkable symptoms?
If the patients are not presenting any remarkable symptoms, that they will be off the radar. But for example, for the study that we have, the NIH Recover study, we want such patients, and we, for example, we have this longitudinal follow-up, baseline 3 months, 6 months, 9 months, 12 months, and then following them once a year for the next 4 years. The goal of that is really to look if there is any panel that can be developed both for symptomatic long COVID and non-symptomatic long COVID. But as of now, all the data that is published so far is pointing to the 4 items I spoke about earlier, which are diabetes is one risk factor for it. If people have diabetes, they need to be watching very carefully. And then the other elements of the panel is auto antibodies, SARS-CoV-2 in blood, as well as Epstein-Barr virus reactivation. And these are just the elements that have been defined so far. I'm 100% sure many other studies will bring up other markers. And the way we know this also is because the virus itself manifests itself in different hosts differently. So different phenotypes of PASC and different subphenotypes of PASC will be defined as we move along.
Okay, thank you for answering that question. The next question is, how are medical professionals being trained in recognizing symptoms of long COVID? This goes to any of you, any of the panels.
So let me take that because I actually see these patients. We have a joint clinic at VA, and I believe we tried to do so in GW as well, where pulmonologist, endocrinologist, and infectious disease specialists actually work together. We have one trial ongoing, as I said, where we are looking at multiple organs that could be affected. Now, unless, as I said, unless you look, unless you check certain parameters, you would continue to miss what is being affected in long term. The reason is prevention of major organ failure, right? So unfortunately, United States practices interventional medicine, but it's now we realize that diabetes and obesity played such a huge role in this pandemic because our preventive measures are fraught with problems. And unless we put this in place now, it's going to break our health budget in years to come. So it's a multi specialty clinic where we all sit together. Sorry, I forgot to mention nephrologists. We also have a diabetes nephrology clinic for the same reason. And remember, these patients that I showed you data on, they were not diagnosed to have diabetes. They may have prediabetes, but in normal situation, these patients would not get anything, right? And some patients would say that, why would I even bother coming? But clearly they are developing kidney disease. Maybe it will resolve. Hopefully, I'm hoping that it would resolve. This podocyte spill, podocyte specific protein spill would actually resolve, hopefully, maybe in 1.5 years, or maybe not. If not, and even that damage that has happened because of inflammation and so on, if maybe we have some medications, if we try those as an intervention, and that's one of our grant proposals to see whether that can improve the outcome. These are some of the parameters. And, for example, ACE inhibition is not just about blood pressure control. It's about blood pressure control, protein leak improvement, may have also improvement in lung function and so on. So these are multifaceted medications that can help. Sorry, that was a long answer to a fairly brief
In
question.
terms of, since you just mentioned this, and you talk about diabetes, we have recently, for the updates of our patients at Howard University, what you mentioned about diabetes and the outcome, we see it really worse in African American with the outcome, diabetes. And
Mhm.
this is really a predictor, and also for kidney failure. We
Yes.
have seen it, which is very important, and I think being recognized, especially for those who were treated either for metformin or insulin. And this is something which we are very interested that I'm sure we can do some collaboration there. Definitely.
Yes. I mean, as you pointed out, you're looking at gut microbiome. So we
Right.
are also looking at what changes in gut microbiome happens to various diets in prediabetes. Why do I keep harping about prediabetes? Because right now this country has more than 80 million people who has prediabetes. Guys, we need to do something about these patients, because even if 1/3 of this population moves to diabetes,
Yeah.
it will break our banks.
Yeah, I think the same things as you mentioned, at least a significant portion is due to BMI. And this is really something which is alarming for kids, because the pediatrician also, the data which is coming, this is a risk factor for many things, including diabetes. And I saw this recently because I'm also in IBD, ulcerative colitis. I saw this is something which is very important in terms of risk factor for ulcerative colitis as well. So you are really touching the very important part of this outcome, especially for COVID now. We have different comorbidity which is coming, and then it is a significant part of the death outcome.
Oh, the next question was, were the participants in this studies vaccinated against COVID, or do you see any difference between the vaccinated and non-vaccinated?
Is this, sorry, let me ask somebody else to comment and then I'll comment. Maybe.
Go ahead. Go ahead.
So vaccination, well, vaccination record, all of these patients were fairly well, very closely followed over the 1 year period. So they had everything that is normally advised. So I believe they would have had the vaccination as prescribed. But as you saw, they clearly did not have vaccine at 10 weeks, maybe not even at 6 months. Maybe some of them did have at 12 months, but there was no drop in protein even at 10 weeks to 6 months, right? Which is very concerning. And though there was, I mean, it's just so concerning that we definitely want to look at what's happening at 18 months. And we have only started to gather some of these patients to see what's their podocyte-specific protein. The podocyte-specific damage in prediabetes, or as we call microalbuminuria or proteinuria, happens way before overt diabetes happens. It's almost the first vascular complication to show up in any diabetes patients, as is hypertension and hyperlipidemia.
Okay. So there's a comment from one of my colleagues here, say, are we shifting our attention to more biomarkers of disease? That seems to be the theme in these research studies. And if so, can we use biomarkers to diagnose pre-diabetics in patients to then apply measures to prevent the development of diabetes?
Right. So clearly, pre-diabetes right now is based on blood sugar parameters. Right? We don't use any other parameters to diagnose diabetes. However, we know that hyperglycemia or persistent hyperglycemia is almost the last stage of this iceberg process. To diagnose pre-diabetes, it's not just about high glucose, it's hyperlipidemia, hypertension, and these are almost always not addressed and missed. Even now, patients who has diabetes, if you look at any tertiary hospital, the statin usage is as low as 50%, or cholesterol lowering. And those are the things that we need to implement so that a virus which uses a ACE2 receptor, which is a blood pressure mediating receptor, doesn't cause devastation. I mean, this virus is very clever virus. It is almost using the same receptors, which is associated with hypertension and which subsequently leads to other vascular diseases. TMPRSS2 is also an interesting receptor. And a lot of work needs to be done to understand the role of these receptors and its subsequent secondary messengers on the vascular system. Yes, we don't, I mean, the long story short, we don't do a good job of diagnosing prediabetes and prevention of prediabetes. Clearly, we don't.
But I just can add some.
One more question.
Yeah, go ahead.
Go ahead.
I think there is a preclinical and laboratory panel, which is now. Can you hear me?
Yes.
I said there is some laboratory panels which is now in the making, and it is not approved at the bedside, but at least for the taste and the genes, which is craving for the food, and turns out to be an attraction for the metabolism, and then working through this pathways. It is something which is now we are understanding more. And with the advancement of technology, I think we will be there soon for the biomarker discovery. But as Dr. Shen said, this is the blood glucose level, and the test which we are doing is the bedside, I guess, part which is significant at this moment. But there is a lot of good biomarker, including mRNA and including some of the genes which is in metabolism, which is now in the making.
Right.
There is a comment here stated that a number of retrospective studies suggest that metformin may be clinically beneficial for patients with COVID. For example, some studies show that lower mortality, improved microbiome, overall lower inflammation. Would you consider starting a pre-diabetic patients on metformin?
Sure. I mean, metformin is approved in prediabetes. It's one of the only medications that has been studied in prediabetes. Right now, NIH sponsored, there's only one prediabetes study we usually refer to, which is, sorry, I'm forgetting the name. But this was published almost 10, 15 years back, right? So the studies on prediabetes, though it affects so many people, what medications actually work and what don't, whether we should use the medications that we use in diabetes earlier, those studies results are not there. Now, some are running. Whether we should put prediabetes patients or COVID patients on metformin, I mean, there is no guideline for it. Now, if we have a prediabetes patient, independent of COVID, we do treat them with metformin, but as our studies showed that lifestyle and dietary intervention should be tried first, or at least in conjunction. As Dr. Ashkotra mentioned, the diet is important, and the worst part is if you look at the obesity pandemic that the country is undergoing, it's not just adult population. The population that has increased significantly in the last 2-3 years or even 5 years is the adolescent age group. I mean, the age group between 12 to 19, that's the biggest hit. These are going to be adults in next couple of years, or they are adults. And again, there is no program in place to recognize and address this group to prevent cardiometabolic diseases.
So
Actually,
do we have any idea how long these long-term COVID impacts will last?
I mean, I've shown you data for up to 1 year.
Mhm.
My preliminary data up to 1.5 years show that it persists. I'm hoping that it's not going to persist in 2 years. I can easily parse out if this is an effect of vaccine, which I don't think so. I think it's an initial assault of the virus. And the fact that we are now told that all of us would get the virus and get it over with, and 60% of adults already had it, and et cetera, et cetera. I don't know what's the long-term health implication of that. So I would still suggest to be cautious and wear masks and maintain all the health precautions that is currently advised, rather than trying to get the virus.
yeah, if
How
I
about-
might just, if I
how
might
about
just
the
add
long-
one thing.
Go ahead.
Yeah, while the focus was here on diabetes, we have really to widen that scope a little bit to metabolic syndrome.
Right.
The same story that happened with NAFLD, which is non-alcoholic fatty liver disease, that is right now. There are proposals to shift the name to MAFLD, metabolic associated fatty liver disease.
Mhm.
So diabetes, obesity, hypertension should be lumped together, and markers for this metabolic syndrome are to be sought in the context of COVID and long COVID as well. I have a vivid story of one family that we had at Howard where the whole family was hospitalized. The parents left after 2-3 days. The siblings left after 4 days. The only person who stayed and unfortunately died is a youngster who was obese. So this is why metabolic syndrome is the way to look for markers for panel of markers, because they are all intertwined anyways. And this is the path to go as to pinpoint to major risk factors or panels to follow up patients.
Last but not last question. The follow-up with the long-term impacts of COVID on children. Do we know the possible long-term impacts of COVID on children?
I mean, there's very little data, as you know, in children, because even the virus or vaccine was not implemented soon enough, I think, in this population. It's just available now. We just don't have enough data.
We do not. As a matter of fact, the project that we're talking about, the NIH Recovered, it has one section for pediatrics. The one we are funded through is for adults. So they have an NIH Recover long COVID for pediatrics, and the data is not there yet. But if there is anything to watch for example, if you noticed the news just couple or 1 week ago, where many cases of hepatitis and severe hepatitis have been shown in many countries. And some of it was associated with COVID-19. The rest is still in question. So that might be something, a major alert that we have to pay attention to. So, and if you remember, I stated earlier that EBV, Epstein-Barr virus, is reactivated in patients with long COVID. There are some stipulation that may be this hepatitis, severe hepatitis shown in very young kids, is also a result of COVID-19 or post-COVID-19 syndrome. So there are a lot to watch, and we have to do our best to avoid major public crisis in the future.
Okay.
Right.
Well, I will thank the panel for great discussion, a great presentation. Thank you much. So our next section will be,
Thank you.
Can we switch over to the next section? Okay, our next question is vaccine and mandate distribution and the structure of herd immunity. The moderator, Mabel, are you on?
Yes, thank you, Jane.
Okay.
Thank you all for the great presentations. My name is Nabil. Kamal Qureshi. I work as a Deputy Conflict of Interest Officer and in the Department of Research Oversight at Georgetown. We will be presenting, we will have two speakers who will be presenting from the Georgetown side. We have Dr. Shweta Bansal and we have Dr. Daniel Salmasi. I will quickly read their bios. Dr. Shweta Bansal is a Provost Distinguished Associate Professor of Biology at Georgetown University, and she specializes in the role of social and spatial behavior in infectious disease transmission dynamics. She leads an interdisciplinary research group that develops data-driven mathematical models using tools from network science, statistical physics, and spatial statistics to study disease surveillance and control in a variety of human and animal disease systems. Today, I'll also read out from the abstract that she'll be presenting. The US COVID-19 vaccination landscape has shaped the population health impacts of SARS-CoV-2 structured the potential for local elimination alongside natural immunity and behavioral containment, and will drive the transition out of the pandemic to endemic circulation of SARS-CoV-2 virus. However, the tracking of vaccination progress at a fine geographical scale and through time has not been a US public health priority. Here we discuss the tracking of COVID-19 vaccination progress in the United States, characterize the spatio-temporal heterogeneity in these patterns, and discuss the epidemiological consequences of this distribution. The next speaker after Shweta, we have Dr. Daniel Salmasi, who is the Andre Halliger's Professor of Biomedical Ethics in the Departments of Medicine and Philosophy, and he serves as the Director of Kennedy Institute of Ethics at Georgetown University. He's a practicing internist and a philosopher. He's the editor of the journal Theoretical Medicine and Bioethics, and has written extensively on many topics in bioethics, especially regarding care at the end of life. His 7 books include Methods in Medical Ethics, The Rebirth of the Clinic, and Physician-Assisted Suicide and Euthanasia, Before, During, and After the Holocaust. His topic will be the distrust as the fundamental unifying theme in 3 pockets of vaccine hesitancy in the US. He will then briefly touch on the ethics of vaccine mandates and the ethics of withholding or deprioritizing medical interventions for the unvaccinated. So I will leave the floor to Shweta Bansal, who can start with her presentation now. And then we can, it can be followed by Dr. Daniel's presentation and then question and answer sessions. Thank you, Shweta and Dr. Daniel for joining us.
Thank you so much, Dr. Qureshi. I'm gonna go ahead and share my screen here. And I'll confirm that you all can see that.
Yes.
Are you all able to? Yes, wonderful. Thank you. Thank you. Thank you all so much for having me. This is a really critical meeting to kind of discuss clinical and translational issues. And I know you all have been talking about COVID-19 most recently, but also other very important issues. I'd love for us to kind of shift gears a little bit, at least for the next half hour while I have you. So to kind of talk about a much more population health perspective particularly on thinking about SARS-CoV-2 transmission, especially from the lens of vaccination patterns. So just by way of intro to kind of add to the kind introduction from Dr. Qureshi, kind of what my group does. I'm based here in the Department of Biology at Georgetown University, and we think most about kind of how social and spatial processes have variation in them, and how we are all sort of connected both physically and socially, and how all those processes really drive infectious disease transmission. And we think about this sort of at the individual scale, both for human populations as well as animal populations. And we think about it at what I call the landscape scale. So thinking about not just individuals and populations, but scaling up these communities to multiple communities and thinking about how each community varies and how it connects to each other and how that might impact our estimation of disease transmission. Now, this landscape scale, honestly, we kind of don't need a better motivator for why we should care about this scale than to kind of look at the SARS-CoV-2 pandemic these last 2 years, right? We can look at the example of these two largest democracies in the world that have, taken a very decentralized approach to public health and to pandemic response during these past 2 years, and we can see that kind of play out on the maps that we've all been kind of carefully watching over the last 2 years, right? We see that geographically there's variation in kind of how intensely an outbreak kind of strikes different locations, and when it strikes has been really variable as well. And as I was just alluding to, right, some of why this variation exists is because of the response that these nations have taken towards the pandemic. The fact that both these countries have very decentralized public health responses is part of kind of why we see this variation, this sort of different parts of the map lighting up at different times. But there's a whole host of issues why we kind of observe and expect this type of geographical structure in disease outbreaks, right? So there's demographic characteristics, there's environmental ones, and then there's a whole host of social structures that we create in our societies, whether they be economic structures or structural inequities, or the public health system structure itself, like really can change how and when disease is spreading in different parts of a community, a country, and the world. But of course, if different locations, different communities were just different because of all these factors, that might be something we could handle from a population health or public health perspective. We would just need an approach that was tailored to each community. But what makes this truly complicated is the fact that the spatial heterogeneity or variation is layered on by a connectivity that exists between communities. So all of us kind of commute from one location to another, maybe not so much in the last couple of years with so much remote work, but in before times at least, right, there's a lot of commuting that happens for both for work, but also for leisure purposes, whether it be commuting by car or public transportation or even air travel. Right. In addition to the physical sort of connectivity that exists between communities, there's also a kind of social connectivity via traditional media, but then now much more so through social media as well. And so what this connectivity amounts to is that this variation that exists between communities, it can really kind of amplify the effect of that variation across a much, much larger scale. And the actions of one community can be sort of felt in far-reaching ways. So these types of factors and this connectivity kind of really lead to this patchwork pandemic that we've observed over the last 2 years. And from a population health perspective, there's a lot of dangers to having to deal with a patchwork pandemic as opposed to something simpler. From a surveillance perspective, we really kind of need to track with sort of what I call a Goldilocks approach, right? We can't track at too large of a scale where we can't just look at cases nationally because we're missing out on a ton of detail, but we also can't just look at tracking of cases within one particular community because we'll be missing a lot of that variation that exists around the country. There's also a huge sort of differences we're seeing with this type of pandemic, with this patchwork pandemic, between kind of true local risk, how many cases are actually spreading versus what is being perceived by the population as outbreak risk. Also, from a response perspective, resources end up being sort of too thinly spread, where at like the federal level, we need sort of continuous availability of resources because there's always somewhere in the country that's, that's in need of resources. And at the local level, we're kind of always reinventing the wheel with respect to decision-making and implementation because there's just sort of not coordination between communities. It also just requires a general agility, which is not particularly characteristic of public health systems, and especially in the US, it's not particularly characteristic of our public health systems. So what I'd like to do with my time with you all is kind of go back to basics a little bit, think about transmission potential at this very, very large scale where we're thinking about what's happening at a local community level, but also simultaneously like thinking about how things are going nationally, and kind of take a pause to think about one specific aspect of this big puzzle, which is vaccination, and then come back to the big picture to talk about how this affects, how that one piece coordinates with everything else to affect transmission potential. So like I promised, I'd like to go back to basics first. So what do I mean by transmission potential, right? At its, most basic, as population health folks tend to have, this quantity called R0, or the reproductive number, that we tend to use to characterize what the potential of a given pathogen is to spread in a population. And, different pathogens, of course, have different reproductive numbers. And with SARS-CoV-2, we've kind of seen this number evolve and, essentially become larger with the different variants that we've been that have been circulating in the population. And R0 is a fairly simple quantity. It's a static quantity, and it's a quantity that really only applies at the beginning of an outbreak. Sort of before an outbreak's begun, it tells us kind of based on how infectious the pathogen is, as well as host behavior. This quantity tells us, how quickly we can expect a wave of COVID-19 or of any other pathogen to kind of disseminate in the population. But this is a static quantity, and really to kind of think about surveillance and response, we need something that's a bit more dynamic. And so R0, this basic reproduction number, has a more dynamic cousin called RT, the effective reproductive number, which I'm guessing is also something you all have kind of come across. And this is basically R0, a product of R0, and takes into account kind of how much of the population is susceptible to a given pathogen. As you can imagine, for SARS-CoV-2, the second piece has been quite complex and changing quite significantly through time and changing in very different ways. So capturing this piece is going to end up being a big part of our puzzle. And this RT quantity that I'll refer to it as RT kind of really helps us think about outbreaks and their shapes and the dynamics of them, right? So we know from theory that at the beginning of an outbreak, so sort of on the left side of an epidemic, peak RT values will be larger than 1. The epidemic will be growing. Every infected individual will be infecting, at least one other individual and more. But on the flip side of that, of a peak of an epidemic curve, RT becomes less than 1. At that point, transmission is still occurring, but it's just slowing down, and each infected individual is replacing themselves, not quite replacing themselves with another individual. It's on average less than 1, right? And so this very simple quantity like really helps us think about outbreak dynamics. It can be helpful in interpreting surveillance data, and it can be really helpful in planning response. But this quantity then, and this one epidemic curve might just represent one community. But going back to our patchwork pandemic and needing to think about this for every individual community in the US, for the entire country, is going to need a more sort of in-depth quantity than just the RT. And so for this, we've been working to kind of come up with a quantity that calculates this RT value for every single community, every single county in the US, right? And just at its core, this quantity is made up of 3 different pieces, very similar to kind of how R0 got turned into RT. We're now kind of expanding this to take into account in information that's specific to that community. So we have something that captures the transmission potential in that community I at a particular time T. We're capturing, how much of the population within that community I is susceptible at any given time. And the point that is kind of critical here goes back to the connectivity point I was making, right? So these communities are different and these 2 pieces are going to capture that variability, but no community is an island. We in fact have this connectivity between communities that can really influence transmission within our communities, and our actions can influence theirs. So we really need to capture that connectivity between this community I and all the other communities that it might be connected to. And there's a whole host of sort of complex social and economic and political and demographic characteristics that influence each of these pieces, but we can kind of summarize them down to things that we can measure, right? So things about behavior in that location and the environment in that location are going to influence transmission potential there. The susceptible population is going to be a function of, how much immunity there is in the population, whether it be natural immunity or vaccine-induced immunity. And the connectivity, again, will come from this physical or social connectivity that I was just describing. So as I promised, right, what I'd like to do over the next little bit is kind of dig down deep into just thinking about vaccination explicitly, since vaccination is one of the sort of important components that'll go into us thinking about this whole equation. And then we'll do a zoom out and kind of think about how vaccination fits into the whole bigger puzzle to help us think about transmission potential at large. And before I go any further, I want to kind of roll the credits here and highlight the fantastic set of junior scientists that I have the privilege to work with. The work I'll be telling you about today includes each of these brilliant folks in either leading the study or having a supporting role in the study. So the first question I'd like to focus on, right, is just, focusing in on vaccination within this bigger transmission potential puzzle. What can we say about the geographic distribution of COVID-19 vaccination and kind of how that tends to vary over geography and time, right? And this is a kind of critical question to answer, right? Kind of answering this question of what is the geographic distribution of a vaccine, across an entire country is an important question that we do critically need to answer specifically for each vaccine and each pathogen. And this is because from what we understand about the distribution of different vaccines across the country, at least within the US, is that it's highly variable. So if we look at influenza vaccination rates across the country at the US county level, those rates and that pattern of distribution looks very different than it might for MMR, for individuals being vaccinated against measles. And if I brought up, 10 other maps for each other of the other vaccines that are either required or not required, right? The geographic patterns will tend to really vary. And so it's not kind of sufficient for us to just assume that whatever the geographic patterns are for other vaccinations are going to apply to COVID-19. There's also a whole host of reasons why there might be differences between different vaccines, and some of that comes from whether they are mandated or not. Kind of comes from media coverage around that vaccine, which might lead to hesitancy, and it just has to also just do with a whole range of vaccine-specific issues. So I know my colleague, Dr. Somasi, will be talking a lot more about vaccine hesitancy, so I'll let him kind of pick up the discussion on that. But the point is, for COVID-19, to understand COVID-19 vaccination distribution, we really need to do the work of looking specifically at COVID-19 vaccination. The other point I want to make is that it's also important, we know from past work with other pathogens and other vaccines, to look at very fine scale data when we think about the geographic distribution of vaccines, right? So certainly pre-COVID, the vast majority of public health data used to either be at the national scale or at best at the US state scale, right? And so these are just two maps that compare influenza vaccination. Influenza is something we tend to study quite a bit in my lab when we're not doing COVID-19 studies. And if we look at influenza vaccination coverage among almost all ages, all eligible ages at the state level versus at a US county level, we see that there's quite a bit of variation that the state level might obscure, right? So just as an example, if we focus in on Washington State, there's some parts of the state, especially around Seattle and Kings County, where vaccination rates are quite high for flu, but farther out east in the state, there's much lower vaccination levels, whereas when we look at the data at a state level, we're kind of obscuring those pockets of susceptibility that surely exist within the state. So this is something we kind of had to have a very clear idea from past work, that collecting these data on as small of a scale as we can possibly do is important, and the US county scale, with there being 3,000 counties in the US, ends up being a pretty useful scale. And so, we definitely decided that we needed to kind of invest in studying vaccination patterns for COVID-19 when the campaign started in January 2021. And it was, of course, important for us to track vaccination patterns because it was important to maximize vaccination for COVID-19. The pandemic was still very much in its early stages. We know a ton more now, more than a year later, but at the time, really, relying on vaccination was our best option, right? But as it turns out, one might imagine that the CDC would be the agency that would track vaccination patterns for the US, but it turned out that with the vaccination campaign starting in about December 2020, January 2021, there was really no data available at a fine scale for US vaccination rates till well into 2021. To about the first 5 months of the vaccination campaign, there was really no information available from the CDC about kind of what vaccination rates tend to be at the US county level. Even beyond that point, so in May 2021, they did release these data, but then even beyond that, that data tended to be quite incomplete, and there were significant number of errors that we found in the data. And so one of the projects that my group's been sort of leading is that we've been compiling since January 1st, 2021, kind of compiling county level vaccination data for COVID-19 from each individual state. So each state kind of has its own COVID dashboard, and in each of these dashboards looks different than the next, and each state kind of releases this information at various scales and various frequencies in various formats. And so we've been kind of collating all of that data, cleaning it, standardizing it, and correcting it from kind of what the CDC is sharing with the country. And so if anyone's interested, you can kind of look at that data at vaccinetracking.us. And it's a product that we're continuing to kind of keep updated. And so from this effort, we were able to really look at the county level sort of geographic distribution of COVID-19 vaccination. And really what this kind of highlights is that there's a lot of spatial variability, a lot of geographic variability in which parts of the country are vaccinated and which are not, right? So most people are used to hearing that 66% of the US at this point has been, completely vaccinated. So with 2 doses of Pfizer, Moderna, or 1 dose of J&J, and 46% of the US has received a booster dose. right? Those numbers really obscure the fact that there's very much parts of the country that are in the 30–40% range, and other parts of the country that are, upwards of 90%, right? So there's significant kind of geographic variability here that's important to capture. And what's interesting about that variability is that it, kind of the patterns kind of emerge in with a lot of patchiness, right? So there's parts of the country with kind of much higher vaccination than expected, and others that are not. What's also interesting is that this pattern's kind of fairly consistent between complete vaccination and booster vaccination, as we might expect. So kind of to dig into these spatial patterns, we also sort of looked at the clusters that emerge from under vaccination within the country, right? These are locations where vaccination rates are significantly lower than expected. And we find that, these happen to be the top 5 clusters, and they happen to all be in the south part of the US, right? These are also clusters, the ones I'm showing you, that are from vaccination patterns in August 2021, but we find that if you look back or look forward in the vaccination campaign, these clusters kind of do persist. And these clusters, again, are locations of low vaccination, lower than expected vaccination for COVID-19. And these clusters matter because our interactions as humans, those interactions that are capable of spreading disease, aren't happening uniformly and randomly, right? We are much more likely to interact with those that live in our geographic proximity. So what happens when you have these clusters of low vaccination, the chances of an unvaccinated individual coming, interacting with another unvaccinated individual are that much higher, which makes the risk of transmission between those two individuals much higher, which also affects the risk of emergence of new variants. So kind of highlighting these pockets of under vaccination is kind of really critical to thinking about, about pandemic response and in general epidemic response. And again, it was really only because we have fine scale data that we were able to do this. We also looked at kind of the temporal trends around vaccination. So each of these little curves that you're seeing in each of these plots belongs to one particular county. And what we're seeing is the vaccination coverage in that county from about January 2021 into summer 2021. And we're seeing these for every state in at least we're seeing them at least for the continental US. And so what we can see is, of course, there's variability that we're seeing across the country between states, but then also variability within states. So like North Dakota has a ton of variation in how different counties, what different counties' rates are. And we already saw that from the map. But what's striking about this, about looking at the data in this way, is that we find a very similar trajectory through 2021 of the vaccination patterns, right? So we see sort of an initial exponential phase from January to about April 2021, where there's high vaccine acceptance. And then, sort of post May 2021, there's a real slowdown in vaccination to the point where there's essentially saturation by the time we get to summer, right? This is, of course, really what's kind of worried us from a public health perspective. And it's really meant a lot of kind of lost opportunities. So in the summer of 2021, there was the Delta, surge. And essentially we were seeing a large number of weeks, looking at the data on that previous slide, we saw a large number of weeks where vaccination had essentially stalled in a lot of part of the country, especially in sort of the Midwest and the middle of the country. And beyond that point, when it was really clear that the Delta wave was emerging and was going to be quite significant, a lot of locations kind of increased their vaccination efforts to increase coverage. But we saw that in about a third of US counties, all the ones that are in blue or gray here, but incidentally, not in the places where vaccination had been stalled the longest. So there was a lot of lost opportunities and kind of catch up that wasn't done during the summer of 2021. And we're still kind of reeling from the effects of that. So to quickly kind of summarize this piece, right, kind of disease and behavior tracking in the US, we find over and over kind of needs to be a bigger priority. The pandemic has kind of brought a sea change in terms of how we do disease and behavioral surveillance for population health in the US, but there's just a ton more what needs to be done in terms of investing in public health infrastructure. And especially with respect to vaccination, there's kind of been the vaccination has been kind of the biggest success story with respect to the COVID-19 pandemic, but under vaccination really kind of continues to plague us, and really understanding the differences, the reasons for that lag, but also kind of the distribution of that lag is kind of critical to moving forward. So in like about the last 5 minutes or so, I'd love to just kind of zoom out from thinking about vaccination specifically and kind of think about how something like vaccination and that landscape of immunity that vaccination builds throughout the US kind of contributes to COVID-19 transmission potential. So for this, I'd like to go back to this notion of this patchwork reproductive number, effective reproductive number that I introduced a few slides ago. And as I mentioned before, right, certainly we need to think about vaccination, to think about what the transmission potential is within a location, but then there's a whole host of other factors that go in, right? There's natural immunity, kind of which variants are spreading at the and circulating in the population at the time, and a whole host of a whole series of host behavior factors that really kind of dictate where the disease is going next. And so really to kind of get a better handle on this, we did this for every US county every week since April 2020 and forward to kind of think about how we can both retrospectively understand transmission potential, but also prospectively understand it. And so we've kind of first built a susceptible profile for each location in the country for every week. Of course, a big piece of this was the vaccination data that I just described to you, where we took that data, but then we introduced a lag to capture the time it takes for the immune response to kick in, and then also the imperfect vaccine efficacy that we've been observing. Alongside that, we also characterized at this very fine county and weekly scale kind of what our estimation was for how many people were infected, or how many rather people had natural immunity against SARS-CoV-2, which incidentally is not as good as vaccine-induced immunity, but it's still important to kind of take the distribution of that immunity into account. There's a second piece to kind of thinking about infectivity. Besides susceptibility, we also need to think about infectivity, and that varies based on masking levels within a community and kind of variant circulation. And as I mentioned, there's a whole host of behavioral factors that matter, that vary by community and kind of how communities are linked to other communities. So we can kind of put all of this information again together to come up with our estimate of this effective reproductive number over time. This just happens to be for one community, right? But it tells us kind of what our expectation is for the epidemic over time. In this picture, where the blue is kind of what our estimate of this value is, and the orange dots are kind of what we actually see happen if we look at data that's where the epidemic or where the outbreak has already occurred. And we can see that kind of starting from the ground up and doing this very mechanistically from first principles still gets us to kind of what we end up observing in the real world. And so having this ability, again, really kind of lends us the opportunity to think about the spatial and temporal distribution of transmission, both retrospectively to kind of understand what's already happened, but also prospectively to kind of think about what might be coming down the pike, depending on how each of these factors, whether they be behavioral or environmental factors or factors around immunity building, that might be changing in different locations at different times. Because this is also a measure that we built from the sort of from first principles, we can kind of break it down in different ways to understand what is the main cause of transmission surges in different communities, whether it's coming from the behavior side or something else. And it allows us to look at, how immune building has the trajectory of immunity building has happened in different communities and how that has influenced their surges. So this is an example of a county in South Dakota versus a county in Vermont, where both of these locations by July 2021 are kind of at a similar level of immunity against SARS-CoV-2, but they both got there in very different ways, where Vermont really built up that immunity through vaccination, whereas this community in South Dakota really built it through natural infections, and alongside with that came a large number of deaths in that community. We can also kind of think, again, retrospectively about how interventions should be implemented, and kind of learning from this allows us to act more appropriately and in more targeted ways in the future, right? So we can think about how there's trade-offs between different interventions. So specifically how vaccination kind of trades off with something like social distancing, where what we're looking at here is that early on in the pandemic, so in spring 2020, at that time, social distancing was kind of really already high. And so if we did have the ability to kind of reduce contacts even more, that would have bought us a big win, because immunity was so low in the population at that point. Whereas later on, when vaccination was much higher, so by spring 2021, and immunity was much higher, social distancing kind of had a marginally smaller effect than, because there was already a ton of immunity in the population. So it really kind of tells us about how different interventions kind of can really vary in their effectiveness over time. This approach kind of really also helps us think about how, again, the actions of one community. So for example, if Maricopa, Arizona takes, solid actions to implement social distancing or increase its vaccination coverage, the blue dots are all communities around the country that would feel the positive effect of that, right? So because those locations are connected very heavily to Maricopa, Maricopa reducing its transmission potential can have sort of vast benefits, across the country. So this is a really sort of important point about kind of one lifting one boat lifts many. So I'll go ahead and just wrap up there, because I think I'm at my time. But really, what I'm hoping that I've kind of demonstrated is that it's really important. You all are kind of doing the very hard work of taking care of patients, and really we need that perspective, and we need to scale up from there and kind of simultaneously think across scales, from the individual patient perspective through the landscape perspective, and really just the collaborations between those different scales are what we need to solve these very hard problems that we're facing at the moment. So I will kind of wrap up there. Thanks again to some fabulous junior scientists that contribute and make this work happen, and of course, collaborators and funding sources. I look forward to being able to discuss these issues with you all soon.
Thank you, Shweta. That was really informative. Do we have any questions for Shweta?
Hi, Dr. Bansal, can you hear me?
I can, thanks.
Awesome. So this is Dr. Sen here. So great talk. So you at one point mentioned about vaccine-based immunity and disease-based immunity.
Mm-hmm.
And do you see is it possible to do a long-term outcome on, let's say, cardiometabolic diseases from those two populations once identified?
Yeah, absolutely. I mean, so there have been great patient populations that have been studied. I think a little bit less so in terms of outcomes so far, but I think that work is coming. But there's some sort of great studies, particularly from Israel, where patient populations that have natural have antibodies from infection versus antibodies from vaccination, kind of looking at outcomes of infection and first of all, just looking at the effectiveness of a vaccine in those populations and then looking at the outcomes. There's been great work there. In the US, unfortunately, we have very few studies that actually look at how many individuals have infection-induced immunity. And so even for our work, kind of putting that information together required a lot of, sort of a lot of stringing together of different bits of evidence. But yes, I absolutely agree with you, kind of looking at outcomes in those populations in the future will be really important.
Thank you.
Thank you. And we have a question by Dr. Salmasi. He asks how one decides the right unit of analysis. For instance, given the variations within many countries, would zip codes be better? You might not have the data, but would it be better?
That's a great question, right? And something we kind of actively are thinking about. So I'll admit, right, that before 2 years ago, even state level data was hard to come by, right? Large part of my job used to just be kind of beg, borrow, and steal population health data at finer scale so that we could consider these questions. But because we've, kind of gotten access to a lot more fine scale data, we've been able to start to consider these questions more carefully. And sort of what we find is that, yeah, the state, and so of course the scale is going to depend on individual countries, but at least within the US, we find that state level is too large of too coarse of a scale. There's just too much variation within states. Zip code is certainly ideal, but it's very, very difficult still to get access to those data. And from a privacy and confidentiality perspective, I don't think those are sort of limitations that we're going to solve. And I absolutely respect the need for data confidentiality. So this is why we end up working kind of at the county scale, because it balances that need to capture variation, but then also preserves privacy concerns, and often means that we're not dealing with a lot of noise, right? That we're getting sufficient sample sizes in our data to be able to make inferences. Thanks for the question.
Yes, thank you, Shweta. I don't see any more questions, so maybe we can turn it over to Dr. Salmasi if he wants to join in. Thank you, Dr. Salmasi. And thank you for joining earlier on my request.
No problem. Let's see if I can share my screen here. Can everybody see the slides? Great. Okay, yes. Good. So this will be very different from much of what you've heard during the course of the day. But I was asked to talk about ethical questions regarding vaccines, particularly with respect to COVID-19. And I've thought before we give any of these talks that it's always useful to say that I think we have an ethical duty to begin with a note of gratitude, that this pandemic was SARS-CoV-2, bad as it was not Ebola. To recognize how much we have benefited from the fastest development of the most effective vaccines in the history of medicine that have such excellent, at least short-term safety. They're not perfect. There are some rare side effects. We can't be absolutely certain about the long-term effects, but overall, this is amazing. And we now also have very effective antiviral therapies. Imagine what this might have been like, but for the state that science is in right now. And of course, we also had Zoom to get us through the lockdowns, and what would have happened without Zoom too. So I think that sometimes we dwell so much on the negative, we ought to say how effective we've really been. Nonetheless, vaccines are a very important part of what we do, and part of trying to get people vaccinated is really valuable to controlling disease and saving lives. So what are the ethical arguments for a moral duty to be vaccinated? I think it's not just a public health argument per se, but there are really ethical reasons that we can offer to patients and to the population at large to be vaccinated. And I'd say that provided there's reasonable safety and efficacy to a vaccine, that we first have a bunch of duties from an ethical perspective toward ourselves, a general duty to preserve our health. Life for all of us is a gift. We don't bring ourselves into being. And each of us ought to recognize that we are inherently valuable. That's our dignity, and we have duties to ourselves to protect ourselves. Now, we also have duties to others, to families, to fellow workers, to the community at large. And then there are special duties for those in special services like healthcare professionals, other first line responders. And so I think there are good moral reasons that we can give for people actually to be vaccinated. But there are multiple possible reasons why some people might shun vaccination, and account for some of that variation that we saw from Shweta's talk. There may be some people who make a deliberate, educated choice. I think it's rare, what are sometimes called free riders, people who will say, I don't want to take any risks, but if everybody else gets vaccinated, I'll be safe, right? There aren't that many people who've done that, and given the high levels that are needed for herd immunity for COVID-19, that would have been a mistake anyway. There are religious convictions that are possible reasons for hesitancy, but that's actually turns out to be fairly rare, that it's purely religious. In many cases, there's inadequate knowledge, fear. Sometimes there's a cognitive bias called unrealistic optimism, in which people think that they are less likely than the average person to become infected. And this is a common reason why people gamble, because they think they're unrealistically optimistic about their chances of winning the pot. So those are possible reasons. It's not as if we don't have any data about this. There are some. Here's at least a study that was published earlier this year. It's limited. It's only data in the state of Florida. But the real reasons that people give for hesitancy are they're concerned about the potential side effects. They feel the vaccines were created too quickly. They don't believe vaccines are effective. They're not personally concerned about contracting COVID-19. They have a general opposition to vaccines. They generalize from somebody having a bad reaction to any vaccine, not only to COVID-19 vaccines. People report allergies to other vaccines in the past. Some people, again, this might be the unrealistic optimism. I don't think COVID is a serious threat. My primary care doctor advised me not to get vaccinated, and that sadly happens. I've already had COVID-19, so I don't feel I need to. And you see very low down, although people exaggerate the importance of it, I have a religious objection to vaccine. Or people saying they have underlying health conditions that prohibit them from getting vaccinated. Politics is often, talked about in this, and there are some variations. And again, this is only from Florida, but I'd point out that it's a little complex. While it's true in this one study, at least, the Democrats more likely to be vaccinated. Independents were less likely even than Republicans. So if people in newspaper reports tend to blame Republicans. There are a lot of independents who don't get vaccinated either. And so the reasons, I think, are more complex than politics alone can describe. Misinformation is often cited, and perhaps there's a role for that. And there have been some statistically significant, although not large-scale, associations between self-reports of tuning into social media channels or radio channels that have large amounts of misinformation. But I think you have to recognize that association is not causation. You can ask whether people are hesitant because they've been misinformed. Well, maybe, but it may be that people who are hesitant seek out information sources that corroborate their hesitancy, or even more likely that some prior mindset, like being green and saying nothing should be inside my body that's not a natural substance, or being socially conservative, would correlate both with hesitancy and with particular information sources and actually wind up being a confounding explanation. My own view is that at the end, mistrust, which is a terrible problem in our society currently, is at the root of most of it. There are 3 different sources of mistrust, and they actually describe 3 different sources of vaccine hesitancy within the population at large. There are a surprising number of people with PhDs who don't get vaccinated, and they are often associated with the folks who think that measles causes autism. They are associated with natural treatments and immunity, and there's a distrust of science from that sort of a population. There's a second population that distrusts medicine as a whole, and that's particularly true of minority populations, and in very particular way, the Black population of this country that has a history of subjection to untrustworthy acts, and so are untrustworthy, are untrusting of vaccines. And then the one that gets talked about the most are those who are distrustful of the government. They tend to be less educated, poor, lower middle class, and socially conservative. What they have in common, what most, I think, vaccine hesitant people have in common is distrust. But with that as a way of background, I was asked to address some particular ethical questions. And the first and a very big one for this country and the world is whether vaccine mandates are ethically justifiable. And second, whether relatively scarce treatments could be withheld or at least deprioritized for unvaccinated persons. And there are people who make arguments on both sides of both of those questions. I'll start the discussion of the first question about vaccine mandates with some general principles about public health ethics. The first principle is a combined one of both beneficence and non-maleficence, that we want by any public health intervention to do good and to limit the harms that we do. We don't want to be maleficent toward persons. That's what that 50-cent word means. We want this to happen both for individuals that we net are helping and not harming, but also with respect, particularly in public health, to the sense of the common good. The population as a whole has been benefited and not harmed. And that's the driving factor in public health ethics. But there are some other principles that are very important that we have to bear in mind. One is transparency, that the reasons for public health interventions ought to be made public in a transparent way. And when that doesn't happen, that begins to build up distrust. So when people are told that they should not bother about masks early in the pandemic, and then they're told the only reason that was being said was so that there could be enough for healthcare workers, people tend to be distrustful of messages they get. A second is reflexivity, that the people making the rules ought to have those same rules applied to themselves. I think we've been pretty good on that one in our public health efforts in this pandemic. A very important one that we'll talk about a lot is restraint, that in order to achieve the public health goal that we're aiming at, we ought to, as a principle, engage the least invasive, least freedom restricting intervention that we can in order to achieve that goal. And the last is a principle of justice, that the burdens and the benefits of any of these public health interventions ought to be as equally distributed as we can throughout the population. just in general, what we're talking about with a mandate is a coercive action by the government. What are the conditions under which that becomes ethically justifiable? It can't be just anything that the government says is good for you can be mandated, right? We have to decide what are the conditions under which it is ethically justifiable. Well, under the principles of beneficence and non-maleficence, we would want to consider whether the harm that's being mitigated is harm to others and not to the individual, right? So if we're saying that we're trying to prevent harm to the population, we have more reason to justify the intervention than if we say that it's only for your individual benefit that we're going to force you to do this, right? That becomes paternalistic, and we don't tend to do that. The harm has to be sufficiently grave to warrant coercion. So we need to sort of say that for the common cold, if we were to have a vaccine, we wouldn't mandate, for instance, massive vaccination for the population for something that's of trivial health consequence. The more people the harm affects, the greater the warrant, and certainly a pandemic fits within that. There's a greater justification for coercion if the harm affects identifiable people and not just statistical lives. And this is a part where there's often debate among the public health public health officials and persons who do clinical medicine because of the difference between statistical lives and identifiable lives. The public health people will say that any life, statistical or not, is a life that needs to be counted, whereas in fact it seems that the justification for coercion seems to be greater when we know exactly who is going to be harmed before we force somebody to do something that would prevent that harm. The harm has to be imminent, the harm should be likely, and the preventive intervention ought to be safe. So before you're going to engage in any kind of coercive public health activity, those conditions, I think, need to be in place. The condition of restraint, I think, is important. And it's important that the individuals themselves are the direct agents of the potential harm, and to say that you need greater justification to warrant a coerced act upon the person of the individual than to prohibit a potentially harmful act. It's a little bit of a philosophical mouthful, but let me sort of say that you need greater justification to force people to eat spinach than you do to prohibit people from using pesticides on the spinach, right? So if it's something you're doing to individuals, you need greater justification in order to justify it. A coercive intervention is more justified if the preventive measure is simple, if it's not invasive, and other reasonable means of achieving that public health goal have to have been exhausted. That's part of what restraint means in public health. And then some principles of justice that ought to be in place is that uncoerced access to the preventive intervention ought to be equally available to all potentially at-risk populations, and the burdens and the benefits of the preventive intervention ought to be equally distributed across society before we start engaging in coercing other people into engaging in some sort of public health action. So those are the considerations in a general way. Begin to think about COVID in particular. I think we need to be clear, first of all, about what the public health goal is in the pandemic. And we vacillated a bit and have, I think, between whether it's to eliminate the virus versus whether it's to prevent severe disease and death, to keep our public health system from being our healthcare system in general from being overwhelmed. Those last two, I think, are very important goals. And whether we're using the pandemic to promote other social goods or whether we're neglecting certain social goods in order to promote public health becomes an important part of what we have to think about when we're thinking about interventions in a pandemic. So here are some cautions actually about a vaccine mandate for COVID-19. I think that there are reasonable reasons to question whether the harm to the general population is sufficiently grave to warrant coercion, and whether we've engaged in a kind of COVID-19 exceptionalism with excessive risk aversion. Certainly there are extremely high risk populations, the elderly, those with pre-existing conditions such as diabetes or immunocompromised, for whom the risk is significant. But for children, the risk is not very great. And so it seems to be that we have approached this with a sort of one-size-fits-all approach rather than selecting the populations that are most at risk. Mandate doesn't typically have that kind of fine-grained look. The harm actually affects statistical lives, not identifiable lives, right? So if I am unvaccinated, who exactly am I actually putting at risk? I think that's a question you can ask. The threat of becoming infected and then spreading is not in any particular case, actually in an individual case, very high. At the population level, it becomes very significant. At the individual level, it doesn't. And the threat of transmission to one other individual is not absolutely at the individual level very likely, although obviously at the population level it is. And the question of restraint, vaccination is a coerced act upon the person of the individual. If you were saying it doesn't matter whether you object or not, we are going to vaccinate you, I think we have to have a very significant warrant for doing that. Vaccines are invasive, and there are other less invasive means such as masking, social distancing, and frequent testing that could be used for individuals who would be resisting vaccination. I'm not certainly opposed to any coercive public health interventions, but I think something that meets those criteria that I laid out more clearly might be if you consider the patient who's actively infected with multi-drug resistant tuberculosis, for whom the threat is imminent to individuals who are identifiable, and the threat is highly significant, and I think the moral warrant, given the kinds of criteria set out, would be much easier to meet for such an individual than it would be for mandating the entire population to be vaccinated against COVID-19. And then the concerns of justice that have troubled me a bit, that we were pushing for mandates for the population when uncoerced acts, at least early in the pandemic, uncoerced access to the vaccine was not initially available to many highly at-risk segments of the population, particularly persons of color, and the burdens and the benefits of the vaccine were not equitably distributed across the society as well. It's important, if I'm going to say that we should be nervous about mandating vaccines, that saying I'm against mandates doesn't mean that I don't want people to be vaccinated, right? I would push for an all-out effort to persuade people. I think it's possible to give them modest incentives. In states like New Mexico, people got $10, I think, for being vaccinated. Things like that might be okay. And we want to make sure that we're balancing public health goals against other social goods like education, economic welfare, social cohesion, arts, and athletics. And above all, I think it's important, as much as anybody wants to control the pandemic and save lives at a population level, that coercion did not and will not help the root causes of vaccine hesitancy, which were and remain fear and distrust. And if people are fearful and distrustful, and then you tell them you're going to force them to do something about which they are fearful and distrustful, in the end, I'm not sure that we're doing those patients or the ultimate public health goals much good. Now, there are some counter arguments. There are people who say we're angry at you for not getting vaccinated. You didn't do your part. But I don't think anger is an especially strong ethical argument. I don't think we can force people to do things for their own good. That's paternalistic. Nor, I think, is there a duty to reshape the population's attitude from a right to one's body that comes out of crass individualism into a moral duty for health. I think we ought to be very cautious about going too far in that direction and cautious about our justifying rhetoric. I mean, this is the kind of attitude toward public health we see in China. Maybe they have been up till the BA2 variant more effective, but it's at a cost. It's at a social, political a cost, and I'm not sure we're prepared for that in this country. So that said, I think there is a special case for mandates in particular populations for frontline healthcare workers who have professional duties to patients and their colleagues and have sworn oaths. I think it's okay to mandate vaccines for that sort of a population. In the military, people who have sworn oaths under military medical ethics. And even among students, there is a principle legally and ethically of in loco parentis, and I think students can, as they were at this university, mandated to be vaccinated. But for the general population, I'm more hesitant about mandating vaccinations, much as I will stand on my head in my own practice and try to get patients to be vaccinated. A mandate is another step altogether. And just quickly, the second issue I was asked to talk about is whether we should withhold or deprioritize treatment from unvaccinated persons. Now, this shifts the focus from public health ethics to clinical ethics, where the principles are similar but slightly different. The beneficence and non-maleficence are now really focused on typically the individual. Respect for persons and their individual choices and freedom becomes very significant. And then the principle of justice is, as I'll say in a moment, important, but compared to public health ethics, assumes a little bit more of a background role in individual cases. So what are the arguments for withholding treatments from the unvaccinated? One is a principle of distributed justice, that others might deserve the scarce treatment more, or that prevention is cheaper than treatment and society shouldn't subsidize the recklessness of persons who didn't get vaccinated and have, for instance, now become infected. There's also an element of what's called retributive justice, or a kind of spirit of punishment. This person doesn't deserve treatment because they did not do their duty and get vaccinated. And that's an argument one hears. Or an argument of reciprocal justice, that they don't deserve care because they will then put healthcare professionals at risk, and therefore should not be treated if they present with COVID-19 infection. But I would point out that all of those arguments, while they seem to be arguments of justice, actually violate the formal principle of justice attributed to Aristotle, that similars ought to be treated similarly. We don't, as a general rule, withhold cabbage from patients who are non-compliant with low-fat diets. We don't withhold ventilators from smokers with COPD. The fact that somebody does something that puts them at risk is not in and of itself taken in any other case to be a reason to withhold treatment from patients. And I think we shouldn't engage in COVID-19 exceptionalism and start doing that in this pandemic. Moreover, we typically don't punish patients for their failure to be compliant with treatments, failure to engage in preventive measures, even as we try to persuade them to engage in those preventive measures or take their medications. What I think we should do is try to understand the root causes of the hesitancy of the person and work with patients where they are at. Nor do I believe that the risk to ourselves is so great that we can't, particularly with the use of adequate PPE, protect ourselves adequately while treating someone who was unvaccinated and became infected, just as we would treat someone who was vaccinated and despite being vaccinated became infected. Because at the bedside, beneficence and respect for persons really dominate our ethics. Generally, if the treatment is effective and the patient consents, we treat. And concerns of justice, as I mentioned before, are typically more background considerations. And they come to the foreground if there is scarcity, but the scarcity of ventilators and antivirals and organs, for instance, is relative and not absolute. And when we allocate scarce resources, the dominant principles we decide on who to distribute them to should be the need of the patient for the intervention and its effectiveness. And those are the frontline criteria in deciding how to distribute ventilators or antivirals or organs. And I think the lesson that a vaccine hesitant patient would learn through our being merciful toward them and not judging them and not blaming them, but actually treating them rather than adopting a spirit of punishment, may be that they actually might learn a lesson and may be valuable to the public health enterprise in the long term by persuading others, don't do what I did, get yourself vaccinated. So the bottom line for me, please don't get me wrong, is get vaccinated, right? And urge patients to get vaccinated. But I think we should continue to use persuasion and not coercion, to tell patients these vaccines are safe and highly effective. They're good for you. They're good for those who love you. They're good for the community. And that the reason we want the patient to get vaccinated is the same reason we would treat them even if they don't get vaccinated, is that we care about them and we want to help them. So get vaccinated. Thank you.
Thank you, Dr. Salmasi. Definitely a very effective message here. And we have some questions. Do you feel any country outside the US approached the COVID pandemic in an ethical manner? What are your thoughts on that?
Yeah, I don't know of much about other countries other than ones that I think are bad examples. I think the Chinese example for me, particularly in terms of surveillance, using patients' GPS signals to track them down, for instance, the kinds of extreme lockdowns, the examples we saw early on of grandmothers who violated COVID going to try to see their grandchild being hauled off into paddy wagons. It's not an example that I think we ought to follow. So I've seen some bad examples. I actually don't know a lot about the rest of the world. I think most of the rest of the world's problem, when you think about the question of justice, has been the fact that most of the rest of the world doesn't have access to vaccines, which is the real looming elephant in the room. While we continue to be talking about whether people in the United States should get a fourth vaccine, a second booster shot, there still continue to be millions, if not billions, of people around the world who haven't had access to vaccines. So from an international perspective, the ethical question is the injustice of our lack of attention to distribution of vaccines around the world. We forget sometimes in our country that pandemic means the whole world, right? Not just the United States or not just the wealthy countries.
Right. And do you think that without mandates, is it possible that the pandemic can continue to rage for a much longer duration?
I mean, I'd ask the public health people on the call about that. I think we have had, large numbers of people who have been vaccinated, right? Particularly those over the age of 65. I think the latest I saw this morning, it finally inched up from 89 to 90% of the population over 65 that's been vaccinated, people at high risk like that. I think we're seeing both because of the high rates of largely voluntary, despite the mandates, largely voluntary vaccination, and the evolution of the virus. I'm hopeful, but others can say more, that we're, but apparently Anthony Fauci did yesterday, that we're beginning to leave the pandemic phase and beginning to enter the endemic. But I guess we have to wait and see. There may be another variant ahead.
Okay, and we have another question which asks about your thoughts regarding mask mandates from an ethics point of view.
Yes, I think mask mandates are easier to justify because of the lack of invasiveness, right? So I think that if someone says that they will not get vaccinated, and then I think they have but less of a justification for saying that despite that they will also not wear a mask, because I think that is less invasive. There's less reason to say that they're fearful, that they can have justifiable fears about long-term side effects, or that this is too much of a restriction on their freedom in that case. So I think that mask mandates are a little bit less invasive, and therefore probably fall below the line that I think we could, in some cases at least, mandate masks.
Okay, do we have any other questions? I think we don't have any more questions. Thank you, Dr. Daniel Salmasi. It was really great presentation and very informative. I think we are 15 minutes ahead of time. So I will ask Sheila to come up for the closing remarks.
Thank you. Sorry. And it was a wonderful day. Thank you all for joining us. Thank you to the speakers for putting in that hard work. And as one who just recently recovered from COVID, I have a special interest in everything that was being said today. And as a reminder, we will send you an evaluation today and again after next week's session. So please fill that out. Please tell your colleagues and your friends to join us next week. If you have any suggestions in the meantime, email me, drop a question in the evaluation. But thank you so much for joining us. And looking outside, there is some sunshine there. So go take a little walk, because no one in your office knows that you're free yet. So you have 15 minutes to go play hooky before you have to get back on your email for work. So thank you, everybody. Have a wonderful day.
Thank you, everyone.